If you are like me and you like to have the content kind of close to your face, you can scan this QR code and it's going to be the deck that we're using for today. There are a lot of, we'll be sharing this deck after the presentation, so if you don't get it now, that's completely fine. But there are a lot of links within this deck, so if you want to follow along and click on some stuff, you'd be able to do this within this link. Additionally, we are going to be recording this session, so one thing I'm going to ask you guys to do, if you do have a question or you want to add to the discussion, there are the mics around the room, and I'm just going to ask very nicely that we use the mics. That way, for people who watch this recording in the future, they can actually hear what you're saying. And again, this is a really, really big room, so if you did ask a question without the mic, there's no way the recording is going to capture this. We will try to repeat the questions, but please, the mics are there. Okay, so the goals for today are we are going to walk you through the journey from data to evidence, which is really what OHDSI is all about. And in order to understand that journey, we have to understand what tools do we use, what are the best practices that OHDSI subscribes to, and what open science approaches do we use. And if we take all those building blocks together, we can actually generate evidence at scale. And we saw a little bit of that yesterday, but today what we're going to focus on are a lot of the building blocks that yesterday we didn't talk a lot about, but are critical to everything that we want to do in terms of generating evidence. Now, for this lecture, I really have two goals, or we have two goals today. First is to expose you to as much kind of the material that's kind of core to what we use at OHDSI, but also what we want to expose you to are all the different aspects of OHDSI. So we want to leave with you that you don't need to be an expert in everything to contribute to this community. And so we want to expose you to all the different parts of what we do within OHDSI. And in looking at that, you could say, oh, I'm really interested in working with data, and I'm really interested in working with, let's say, image data. There are people within the community who are thinking about that. So you don't need to contribute to all parts of what OHDSI does in order to generate evidence. You can find your little area that you're interested in, and there's probably people in the community who are just focused on that little bit. So part of today is to expose you to everything so you can start to see, well, where do I fit in? So today we have a couple of faculty. My name is Erica Voss. I'm a senior director at Johnson & Johnson. I'm in the global epidemiology organization within Johnson & Johnson. We have Christian Reich. He is the CEO of Odysseus Data Services. We have Fan Bu, who is a postdoctoral researcher at the University of California, Los Angeles. We have Martin Lavallee, who's a data scientist at Odysseus Data Services. And we have Marc Souchard, who is a professor representing VA informatics and computing infrastructure within U.S. Department of Veteran Affairs. This is our agenda for today, and so what we're going to do, I will start giving you a very high-level overview of OHDSI, just so we're all on the same page in terms of our history. And then Christian will start in talking about data standardization. So you probably heard a lot yesterday about the OMOP common data model, the vocabularies, but we didn't talk about any of that yesterday. Christian's going to introduce that idea so that will make a little bit more sense. Then from there, Fan will talk about methodological research and really kind of our core pillars in that. So in OHDSI, we talk a lot about characterization, we talk a lot about population level estimation, and patient level prediction. And Fan is going to give you an introduction to those three pillars. From there, we'll take a quick break. I believe there'll be coffee and potentially food, but hopefully we'll re-energize at that point. We'll come back, and Martin is going to walk us through open source development. And I actually felt bad for Martin preparing this because there's so much that's getting developed within the community that it's really hard to be able to like showcase everything within 45 minutes. So all Martin can do during his 45 minutes is really kind of show you kind of the key tools that we think about within OHDSI and use, as well as kind of explain like how we do open source development. Then from there, we really have the building blocks that we need to build evidence or generate evidence at scale. And so then Mark can talk about, well, once we have all these building blocks, what's an example of what we can do with that? And then from there, we'll just conclude the day. So that's where we're gonna go today. So I'll start with a very brief introduction. So OHDSI is the Observational Health, Data Sciences, and Informatics. And like George said yesterday, we're almost at about 10 years. So we were founded in 2014. And we are a multi-stakeholder, interdisciplinary, open science collaborative to bring out the value of health data through large-scale analytics. And I really like this statement because it talks about the multi-stakeholder. And why that's important is that we are not generating evidence for one group of people. We don't just care about answering questions for pharma. We don't just care about answering questions for hospitals or patient groups. We care about answering what are the critical questions that need evidence generated. And I think what's interesting about that is it allows us to be open to lots of different questions, not just thinking about one kind of narrow viewpoint of what we need evidence for. We're interdisciplinary. No one individual or group could do this work. We can't just have a whole bunch of statisticians and hope to solve these problems. We can't just have a whole bunch of computer scientists and hope to solve these problems. We need to bring together lots of different types of people, and just kind of like we did yesterday where we're solving the puzzles that Patrick put together for us, we really need to all come together in order to complete the tasks that we want to complete. And the only way that we can come together is through open science. It's not feasible for all the different type of people that you need to be in one organization. So we need to be open in order to be able to collaborate. So this allows people, I'm in Johnson & Johnson, so I'm in the pharma industry, but then through open science, I can then collaborate with my friends and colleagues in academics or at different institutions. OHDSI's mission is to improve health by empowering a community to collaboratively generate the evidence that promotes better health decisions and better care. And that's kind of what drives us and what we think about when we're trying to answer new questions. There's a little bit of hinting at this yesterday, but we are an international community. So the global symposium is here in the US, but there are symposiums that happen around the world. So regularly we do have a European symposium, and we do have a symposium in Asia Pac, but there are groups all around the world. The image I have on the slide is last year's image. So the new Join the Journey book that everybody got yesterday has the new image. We're almost at about 4,000 collaborators at this point. I think it's like 87 countries or 84 countries at this point. We're still in 21 time zones and 6 continents, but we are one community. We do need a central coordinating center to kind of have one place that gets us organized, and that's not far from here at Columbia University in New York City. Now, you probably have heard everybody say the word OMOP and are wondering where that comes from. So I'm just going to give a little bit of history of where that comes from. So OHDSI has grown out of a project called OMOP, and that's the Observational Medical Outcomes Partnership. And this group was very different than what OHDSI is in that it was a public-private partnership established in the US, and it was formed for a specific reason, it was formed with specific people, and it had a specific goal. It was a 5-year project that was looking to establish new methods in observational research and try to understand how we could use this data better to answer questions specifically about the effects of medical products. And one of the things that came out of that was the OMOP Common Data Model, and Christian's going to be talking about that in a little bit. But it was a project, so it ended, and when it ended, that's when OHDSI was founded. So I just want to make sure everybody knows that's where that word OMOP comes from, and because we had already really invested in the Common Data Model, we didn't want to change it to the OHDSI Common Data Model. It really came from OMOP, same with the OMOP vocabularies. Okay, so some of you might have done some homework to read this paper. If you did, great, you get an A. If you didn't, that's okay. We're going to talk through it and you'll be able to follow along. This paper is kind of be the backbone of our conversations today. And throughout all our examples, while we talk about databases, methods, and development, we're going to kind of tie it back to this paper. So it's just a running example. But I'm going to give you a very, very high level overview so that you hopefully it will help you follow along. And actually, I do believe George talked about this a little bit yesterday. So this was published in 2020 by George Ripsack et al. It's in JAMA Internal Medicine, and it's the comparison of cardiovascular and safety outcomes of chlorothalidone versus hydrochlorothiazide to treat hypertension. And so what's just really important to understand now is that chlorothalidone is currently recommended as the preferred thiazide diuretic to treat hypertension. But there's really no direct evidence like from trials that say that compared the risk and benefits, although I guess technically now you could say there is because there was a trial that came out after we did our work. And hydrochlorothiazide is a commonly prescribed member in this class as well. And so OHDSI set out, well, let's compare these two and see if we can come up with some evidence that suggests how people should be prescribing these drugs. So we had two exposures that we were interested in. And Fawn will talk a little bit about when we set up a population level estimation, we'll have our target and comparator, and then we're typically going to think about those target and comparator in a set of outcomes. In this paper, there were some primary outcomes like acute myocardial infarction, hospitalization for heart failure, ischemic or hemorrhagic stroke, and a composite cardiovascular disease outcome. So we're going to compare these two drugs to see which one has higher risk of experiencing one of these outcomes. Now, we talk a lot about building blocks, and if you read the paper, I'm going to just highlight some of the building blocks that went into generating the evidence. So first thing, you need data. And so in this study specifically, we used two US claims databases. That's the MarketScan Commercial Claims and the Optum Clinformatics Data Mart. We also had an electronic health record. That was the Optum de-identified electronic health record. Now, if we were to use these in our native format, we would have had to write 3 separate programs, and that would have taken a lot longer. So Christian will talk about how we standardized the data to use the data a little bit more efficiently. Additionally, another building block that we need, and we talked about this yesterday as well, is we need phenotypes. So we need to think about how are we actually going to go find the people of interest. So we need to go find people who are actually exposed to our drugs, which sounds simple, but you need to think about what's the logic that's actually going to go find those exposures. And very similarly, and probably more complex, is how do you find people who've experienced the outcome? And so coming up with the ways and the code to go do that. You also have to think about what goes into your study design. So OHDSI has a way of standardizing the way it thinks about pulling together what you need to know in terms of study design and being very clear and explicit about what choices you've made. So things like what's your target, what's your comparator, what are your outcomes, and what's your time at risk, just things like that. But once you've put together your study design, we want to use the best methods we know in order to solve and answer the questions. So Fawn will talk about things like propensity scores. How do we come up with a way to make comparable groups to compare them for our outcomes? And we also subscribe to doing empirical calibration with negative controls to help us understand residual bias within our data. And we have tools and methods for doing that work in a standardized way. Additionally, Martin's going to talk through some of the standardized tools that help us answer this. So for example, within this study, we could have used database diagnostics to look at our databases to understand are they of high enough quality to actually be used in this question. We can use cohort diagnostics to evaluate our phenotypes to see, well, given my databases, do these phenotypes even play out well within our databases? Atlas is used for many, many things like building our phenotypes. You can put together your study design. It can actually generate R code to help you automatically use all the best in class methods that we subscribe to. So Atlas is very powerful in facilitating you using our tools. And those are just some of the building blocks that come together that allow you to then generate clinical evidence. So that was a very quick overview. And now, Christian Reich, you're going to talk about the backbone, I think, of OHDSI, which is the OMOP Common Data Model.
Thank you, Erica. Actually, we can't walk up and down because there's only the microphone here. Okay. Usually I run up and down. Okay, we are talking about data, or generally standardization and harmonization, why it's important, why we put so much effort into it. So here's a little bit of a cartoon version of explaining why that is the case. So the traditional approach of how you do research in this realm, observational research, is the following. You have, let's say you have a question, can I predict the outcome of patients with renal hypertension? Any question. Okay? And this is like an appliance that gets plugged into the data. Okay? And the problem is everybody's data looks different, always. Doesn't matter that it's always the same thing. Patients have a problem, go to the doctor diagnoses, treats, prescribes drugs, patient gets better, patient doesn't get better, comes back. All over the world, the exact same machinery, the same principle, and yet every single institution thinks they need their own representation how this actually happens and what works. Everyone, there's not one institution that has the exact same data representation than the next one. Okay? So we have this problem, and what do people do? They wire it up. Okay? They build a little script for the data that they happen to have in their organization, whether it's at EHR or a data warehouse, whatever that is. Okay? And obviously, it works. But you have no transparency, it doesn't scale. Those people, they are expensive, they're hard to get. This is not the way to do this. Okay? And obviously, I use this metaphor, this comparison, because if we have a standard, we stop even thinking about that. If you buy any appliance, an iPhone, whatever, you go home in the expectation you can plug it in and it's going to work. Okay? There's not even a question. And so what should happen? Of course, this. We should have a harmonized way how the data are represented in our data stores so that a tool can plug in. And not just this tool, there's a whole lot of other tools. In fact, a little industry can grow and come up, private or public or open source, who makes really good appliances. That's actually how it works in the electricity world as well. Okay? The appliance is built, designed, and sold completely independently from how you get the electricity out of the outlet. So these are the different other applications or use cases, as we like to call them. So we call those the OHDSI tools, and this the OMOP CDM. Why is it OMOP CDM? Well, because when OMOP was there, it was the OMOP CDM, and then nobody wanted to rename it because it's a lot of explanation. Is that the same thing? So it's just OMOP CDM. So today, people really use the words OMOP and OHDSI pretty interchangeably all the time. Don't worry about it. The words now have its own meaning. Nobody actually knows what they stand for. They're a verb now. Have you OMOP'd your data? If you have this situation, analytics can be remote. This line can be very long because it doesn't matter anymore because it will work. I can come up with a tool or a script and send it to Korea and they're going to run it and it's going to give me the right results, except they are bugs. The analytics can be behind firewalls. I don't need to have the data in hand. They can be far away, and I just send the analytic over. So the analytics comes to the data rather than the data to the analytics, as it is done traditionally. And before you know it, you have a network, and we have a network. We have a network of 565 or something like that, data partners who declare that they are data partner. And then there's another unknown number of those who don't declare that they are data partner, they have data in OMOP, but you will stumble over them all the time. You're going to talk to somebody and saying, you know, OMOP, oh, we have data in OMOP. And then I go, why don't you come? And we do things together. So that's the secret sauce, or one of the secret sauce, why this is so successful. And you've seen all the maps and the statistics of OHDSI. So it's not just, so what actually is it? What's the outlet? What can you standardize? You can standardize a whole lot of things. It's the data structure, the data content, how you represent it, the data semantics, what it means, what's a bipolar disease, definitions of cohorts, these are the populations that we are studying, covariate constructions, we need that for the statistics, because everything is always connected with everything. You need a lot of statistics to get the effect of the covariates figured out, whether it's actually relevant to what you're studying or you should remove that effect from what you're studying. There are tutorials for all that you can see. Analysis and results reporting and visualization and dissemination and all this kind of stuff. All this can now be standardized. Okay? So let's talk about the CDM, which is standard really to the data, the content, and the semantics. Not the other things, there are other tutorials for that. But the same idea. Look, this is a patient. We've seen those summaries yesterday when Patrick and Anna and Martijn showed us in the main session the ability to adjudicate or verify. So this is what it looks like. There are these little chunks of texts. That's what doctors dictate and then send as a letter or electronic message to another provider who's seen the same patient in order to know what's going on. Okay? That's a text. That's really where the data originates, in the head of somebody who knows what's going on. And you can't really do much with that, except ChatGPT. Computers are not good at reading this and knowing what it means, and we don't even know whether ChatGPT really knows or what it does. You need to have it as data. It's proper data. Why do we need this information in data? There are two reasons. One is we need to build software. The other one is we need to be able to share data. Share doesn't necessarily mean they have to move, but at least we have to be able to give it to somebody, even maybe remotely, and they know what's in there. Okay? This is not us, this is everywhere. Okay? Every application needs a data model, so where the data sit, and so that's the context which understands what's in there. Same here, and so we can't be without that. And the better your data model, the better your information systems are going to be, and the better your data sharing will work. And obviously, data sharing is very important to us because we are a network. And so, all these, if you take these discharge summaries, there are tons of facts in there. And that's what constitutes the data. And so if you really think about it, what patient data look like, it's like a diary, okay? It's like something happens to a patient at a certain day, interacting with the healthcare system somehow. Most of the time patient comes to some kind of a healthcare site. Sometimes somebody visits at home, but that's actually not very common anymore. But it's still an interaction, a healthcare interaction. So there's a healthcare interaction. We're not interested in what the people do otherwise, outside that. And things happen. Something is wrong. Okay? And then something happens, either the diagnostic procedure or therapeutic procedure, and some drugs are taken, prescribed, administered in various different ways. And then the next one, and then the next one, and that's one. This is patient data. That's it. Everybody knows how a patient works, because we all are patients at some point. And these are the data. So, and what you see here is a flat file or a flat table, which means in a row is something that belongs together. You're not really told what it is, but you kind of like can guess it from what's in the row. Okay, so everything which is in the row is related. Everything which is a column is the same kind, but it is not related. It didn't happen together. So these are the flat files. Everybody understands them intuitively, except the computer. Okay, it's very simple, but not all the data fit in there. So where's like the guy is 59 years old? Where's that information? Doesn't fit here, because these rows don't contain that information, and they shouldn't, because the age doesn't really change from October to November, at least not for this guy. Okay, and and these things here that are in the columns aren't really defined. And now you think about analytics. So this is one patient. This one here is a couple months. Okay. So now imagine we have not one patient, but we have 160 million. There are those databases, and it's not 2 months, but 35 years. So then you immediately understand that using flat files isn't really a good way to store the data for analysis. It's not a good way to store the data just for storing it, because if something new happens to the patient, you have to add it. So where do you add it? Do you add it where the patient is? Do you add it all the way at the bottom? If you want to change something, you have to get in there and start. Flat file is not a good way, but it's very intuitive, very simple. And there's, for example, bioinformatics, and that's what they do. They're not into they're not necessarily gravitating immediately into more complex data structures. I already said that. Hard to build, hard to maintain, hard to analyze, and you don't know what, you can't actually put everything in there. So you need a model. You need a data model. What's the data model? It consists of two things, the objects and the relationships. Okay? Objects are the different things that we have. For example, the patient itself, birth date, sex, and then, I can't see this, which I can look at there. The visits, and the diagnoses, and the procedures, and the drugs. So anything that you saw as a column really is an object. Well, some people define slightly different objects than others, but the principal idea is that the type of entity that you want to describe goes into one piece of information, an object. And then there's a relationship, which means this patient here, 1534-044, belongs to these visits. Okay? So you have relationships between these objects. That's a data model. You can, usually you represent it either as a logical model, that you actually show the various fields or attributes, and then you make little lines between them to say, look, this is the patient ID, and it goes to the patient ID. Okay, so these things belong together, it's the same. Or a conceptual model, where you're saying, look, it's a higher level. I cut this patient data into these 5 different objects, and they belong to each other. And patient is the one that unifies them all, where they all come together, which is what this little bubble thing here means. Or the physical model, which is computer language to tell the computer this, a little harder to read for people. So when you hear this is a physical model, conceptual model, you don't abstract from all the details. And the logical model, where you have the details and you give the logic that's happening inside. No data. The model doesn't contain data. The model just describes the model. What else? The data need to be normalized. We do that in OMOP data model a lot. Other data models that also exist do that too. What does it mean? Well, there are certain principles how the data are organized. For example, you don't want duplicate records. So, for example, this one here is a duplicate, at least partially. Okay. So I'm talking about 1st of November, same visit. Obviously, he didn't have 2 cystourethropscopies. Okay, this guy had only 1. It's actually nasty enough. Nobody wants to have 2 in a day. And we know it actually is it only happened once, and this is a duplicate. We can tell that. You want single value per field. So anesthesia is given. You don't have to give it for the cystoscopy, but it's really nasty procedure, so given to this particular patient, and the cystourethropscopy. Okay? So what you do is you don't squish several things into one field. You put one thing in a row. Okay? And then, of course, you have to repeat the date. And so same as for the drugs here. So it's like the whole cocktail in this anesthesia procedure, a little less in this one. And then so you repeat the 1st of November, and you repeat the 12th of December. No transient keys. So look at this. Fentanyl, the official one, not the illegal one, is an anesthesia drug. Okay, it's actually been there for a long, long time. And the fact that it's an anesthesia drug is independent from the fact that the patient got it on 1st of November. It has nothing to do with this. So split those things up. Okay, don't have them in one data set, in one data record. It's true, but really what you want to do is you want to say, well, fentanyl is anesthesia, always is. I have a table for that, which is the drug class table, and I tell what the patient actually got. Okay, splitting up the information that happens through transient key. The transient key here is fentanyl. The real key is date and patient, and the transient key is fentanyl. Don't use lexical terms. Don't write cystourethropscopy in your data. You can, but actually if you Google cystourethropscopy, you get to a Wikipedia page of cystoscopy. So what now? Which one is it? Well, it's actually the same. Okay, usually, nobody actually, I don't know why CPT4 calls it cystourethropscopy. Everybody says cystoscopy. So, but officially it's a synonym. And synonyms and jargons and abbreviations are all over medicine and healthcare. You will die if you do verbatim data. Don't do verbatim data. Code it. That's what practically all systems do, but to a different degree of how much they code. Just the things which are really overwhelming, or do they still have male and female as little words? And you'll get that, okay? And so what do? You actually create a concept, an identifier, or a code. Many people call it codes, and officially it's also often called coding system. And there's a profession called medical coder in the hospitals, and what they do is they go through the records and say, oh, this is this one, because they have to submit this to the health insurance and the government, the payers, and the payers want the codes. They don't want the text, for good reason. Same reason why we don't want it. So what you do is you then have a separate table where the code gives you the name, may give you other attributes, and the synonym. Okay. And then you have outsourced this problem. This is called the reference table. It has nothing to do with the data. You can even make it public. Okay. It doesn't mean anything. It just means these are the things I'm talking about. But they're in one place and they're really kept clean. So here's a data model, OMOP data model. That's it. You have to now take a photographic memory of this and know it from today by heart. You actually, if you start working with this, you will ridiculously know it very well by heart at some point. You know how these, like all the names of the fields and everything. But at the beginning, that might look a little overwhelming, but it isn't. In fact, there are other data models which are much more complicated and messy, for good reasons and bad reasons. So everything is around the patient, person called person, because again, there's actually no difference between persons and patients. So that's one way to normalize, that you pull together all the data that belong to one patient. And here's our reference table. And everything that happens to the table in OMOP has to be a concept. There's no verbatim, almost. There's a little bit of an exception, which I'm trying to get rid of. But for now, assume there's only everything that's in the data has to have a concept in the table. Otherwise, it's not there. Okay? And that's very important because we're going to talk in a minute about it, because we all share the same reference. Part of the OMOP model is that everybody has to use the same reference. You can't make up your own. You actually have to come and say, I need a code. And then some people will say, really? Show me what you got. So that's painful, but it's a prerequisite for this to work. Question is, what's the difference between location and care site? Location is just any location, for example, East Brunswick, New Jersey. It could be where a patient is, where the care site is, where something is, just a location. Could be like a longitudinal, latitude, longitude a combination. And care site is an institution, healthcare institution, so brick and mortar. Okay. A virtual visit probably wouldn't have a care site because, there's no care site. But if you're going to a hospital, I don't know what the hospital is called here, around here. Huh? Robert Wood Johnson Hospital is a care site. Okay. Or Dr. Jones' office is a care site. So that's where healthcare happens. And provider is flesh and blood. This is brick and mortar. This is flesh and blood. You're going to run into this. There are data models, which in the US particularly, which mish and mash them together. Okay, so a provider could be a person and could be like a group of doctors or nurses, or could be a whole big fat hospital. We split them up. These people actually do things, and they have specialties, and they went to schools, and these have doors and windows and that type of thing. Patient-centric, we said that. Here, the blue stuff is actually what happens to the patient. That's the flat file that you saw. This is what happens to the patient. And that's actually more than just drug, procedure, and condition, even though they're the most important one. Well, there are taxes, but we don't have that. There's death, visits we did talk about, devices, measurements, also known as lab tests or other tests, observations, just general things that come up, and so on. Okay. Something else? No. There's health economics. So if you want to know what things cost and what the cost outcome of a certain intervention is, if introducing GLP-1 inhibitor agonists or Ozempic, what type of cost implication does it have? How much does it cost people to routinely take it. Okay? And then the healthcare payers, either the government or the insurance companies. And then there's the exception with text. There's a table called Note where you put those blurbs in, those texts in. But usually the purpose of it, to have this in OMOP is to actually run computer programs called NLP, natural language processing, to get the stuff out and have it in a structured form. So many people have nothing, most people don't populate this because it inevitably is not anonymous information, because it talks about real people, either the providers or the patients. And then there are derived data which are for convenience, so you don't have to derive them each time you do an analytic. And these are the vocabularies, or the reference, or the value sets, or the ontologies. They're all synonymous words that people use. In OHDSI, we call them OHDSI vocabularies, but the communities outside, they're not here yet. They call them other things. They call them value sets, which is, for example, a term that FIRE uses, ontologies. They're probably not other words still. So again, here are the concepts, and here are our relationships, and then there are other things which just are references to the reference. One thing I want to, last thing I wanted to explain here, because it usually does not get covered. There is a knowledge representation theory, and they define formal logic as a open world assumption or closed world assumption. What does it mean? Open world assumption means what we know is true, what we don't know, we don't know. Closed world assumption is what we know is true, and what we don't know is false. That's the only difference. It's a huge difference. What does it mean, what we don't know? Well, it means what's in the data. Do we have a record or not? Okay. So here's our patient. You know, we know there was a visit here, ambulatory surgery. Bladder cancer was the diagnosis at this day. And then it is cystoscopy. We know this. We assume that happened. If not, it's a quality issue, and somehow you need to either flag or fix that. But the assumption is, if it passes quality, all I can do is believe what I get. Okay? So we believe these to happen. What we know is true. Assumption number one. What we don't know, which really means we have no record of, that day is patient didn't take a drug, or did patient take a drug? Did the patient have another visit at that date? Because we don't have a record. We only have this record on this date. Okay? And what was the diagnosis? Was there something else going on? High blood pressure? Ingrown toenail? So the question is, that isn't there. Are we allowed to assume that it didn't happen? Or should we assume, well, maybe it was? And why is this important? Because that's how we do the analysis. If we count rates, okay, incidence rate, prevalence rate, so how often do things happen, either to a population, to a patient, we take this table and just say, okay, how often does this guy get cystouroscopy? Could have copied it. 1, 2, 3. Okay, in 2 months, 3 cystoscopies, the poor thing. If we believe that we can trust that we are not, that the fact that how should I say that? We have to trust that if there is no record of another cystoscopy, there was no other cystoscopy. If we don't know this, we can't say there were 3 cystoscopies in 2 months, because there could have been 4. And so patient data, the way we use them, observational data, have to be closed world assumptions. And it's very important because there are a lot of data that you will encounter where that's not the truth. There's a survey. The survey didn't even ask about cystoscopy. What do you do now? Can you run the incidence report on cystoscopies in a survey? No, you can't, because nobody asked, so you don't know what the answer is. It is very important that we are clear. So people, they implicitly assume this. When they do analysis, they assume this. But then people have data, and they don't assume it, and then you get into conflict. And that happens all the time. If we had an open world system, we wouldn't know whether a patient took a drug, had another visit, or had another disease. And that gets violated all the time. And I'm giving a little bit of a pep talk here, that you guys have to fight it and deal with this and talk about it and saying, guys, this is closed world assumption violation. Negative records, okay? People have they want to put in didn't happen or don't know or any of these kind of things. It's not closed world assumption data. No timing, In a survey, did your doctor tell you you have high blood pressure? When? How do you do research if you don't know when the high blood pressure happened and whether that's an effect of another drug or whether your drug fixed the high blood pressure? You don't know that. You can't say that. Medical contradictions, prostatectomies in female patients. Okay, so obviously that's a violation to the first assumption. You have a female patient and you get a prostatectomy. Data with infinite or near infinite possibilities. Okay, so genomic data are not closed world. You don't have a list of all possible things that can happen, all variants that can happen. There are actually billions and trillions. And we don't have a list, okay? Open world. You cannot run an incidence report on genomic data the way you get them. We can convert them, that it works. That's it. But not the way they come. Free text, obviously, you can't do anything with that. Survey data are very often violations. So if you get survey data, you have to be very careful. Because the usual tools that OHDSI has will get in conflict with that, often. There's some registry data and survey data which are good. Clinical trial data, same thing. People saying, can I convert clinical trial data to OMOP? Yes, you can. You don't have a closed world assumption. You don't know what you don't know. That's a big problem. Okay, last one. I'm probably out of time. How much? What? Okay. The vocabularies. So this is the reference. So a cystoscopy has to be in a table. There should be one record, and it's called cystoscopy with a synonym cystourethropy. Okay? That table is called concept. Everybody has to have that table. All the other tables come from your data. These are the patient data that you have that go into the other table. The concept table comes from OHDSI. You must use the concepts that are given to you by OHDSI. Okay? Everybody has to have the same concept table. Why? If I want to run a study like, give me all patients who had 2 COVID vaccines and the breakthrough infection afterwards, COVID-19, I write a query. The concept ID for COVID vaccine, whichever it was, and you make a list. And COVID-19 has to be the same no matter where, okay? In a database in New Jersey or in Korea or in Italy. Otherwise, I can't run the study. And somebody in that institution has to say, oh, what is it? It's like putting a little man into the ATM machine to count the money. It only works, okay, if the data model has a standard representation of the data, which means you can't be the one who defines what the standard representation is. It's the standard, it's OHDSI. It's the only thing which OHDSI really is brutal on to tell you that you have to use the concepts. And then there are relationships, which are also given. And then there's a concept ancestor table, which gives you hierarchical relationships pre-calculated. And then there's source to concept map, which is a table where you can have mappings. I'm not going to go into that system. So what are these vocabularies? Yep, question.
Are those tables fixed, or do they evolve and grow as we get new data types and new technologies?
The question is, are these tables fixed, or do they grow and change over time with new technologies? Yes, they change over time, which means you have to keep getting the concept table. So here's how it works. So what is actually in there? In there are a whole bunch of these concepts, these codes. Where do we get the codes from? We steal them. Because actually we're not the first one who have that problem. The data, the source data where your patient get the EHR or so, where your patients are in, they already use coding. And the coding is usually prescribed on a national level. Sometimes it's not just one coding system, it could be several. But I don't know, in the US, people use ICD-10-CM. They always say ICD-10, which is actually wrong. It's ICD-10-CM. The ICD-10 is 20,000 diseases. ICD-10-CM, the Americans have 110,000 diseases, a lot more diseases. But of course they don't. It's just more detail in those concepts. And so what we do is we just import them and put them in one representation. So you don't have to do this. There are lots of them. There are 140 vocabularies in the system. Not all of them are public ones like these, but many of them are. And you don't have to take these, download them, and convert them. That's a pain. And you would have to do it all the time because they change. So that's done centrally for you. You just rely on a team who does it. Alex is sitting there, who's running the team, to do that for you. Okay, sometimes there's a mistake, but generally this thing has been stably changing since 2009. Okay, and if something didn't change, I don't know, myocardial infarction, the concept stays from release to release to release to release. If it does change, there was no COVID-19 in 2009, it gets added. So there's a constant flux, which is painful because you have to keep downloading and reapplying the concepts to the reference tables to your data. But it's inevitable. That's the world. So essentially, think of a concept table as one long table of all these different vocabularies in the same format, stacked on top of each other. And, their vocabularies and their vocabulary, they have IDs. And the problem is the same thing gets said, that gets repeated in different vocabularies. Every country has a way to represent diagnoses. And of course, COVID-19, each of them have COVID-19. So we have like 20 COVID-19s. That's not normalized. We should have one, okay? OMOP is the only one who says, okay, yeah, we agree. And what they do is they pick one of the COVID-19s and make it the one that's more equal than the other, and the rest get mapped into this one. So for diseases, it's usually SNOMED that gets to be the standard concept, as it's called. And all the other guys are mapped. And says, if you have in your data an ICD-10 COVID, map it over to SNOMED. So now you're saying, I have to map all these codes? That's crazy. It's like thousands and tens of thousands of them. No, you get that together with a download. All these mappings, maybe not all. If there's something missing, you have to complain. But most of them are there. Okay, so you just apply it. You say, okay, all right, here's my mapping table. I just throw it onto my data, and it's going to give me the standard, and the standard goes into the record. So everybody is on the same COVID-19. So we get away from the fact that, the same meaning is represented in many, many different ways. Okay, we have to have one way. Everybody has to speak English, or some kind of English, in the OMOP CDM. Not going to have time to go into this. Don't think of diseases as like independent from each other things. They actually have a lot of relationships to each other. Most important one is hierarchical. Okay, so atrial fibrillation is a atrial arrhythmia, and a fibrillation, which is a supraventricular arrhythmia, which is a cardiac arrhythmia, arrhythmia of the heart, which is a heart disease, which is a disease of the cardiovascular system. So if a patient has an atrial fibrillation, doesn't have 1, 2, 3, 4, 5, 6, 7 diseases, has one disease, okay? But sometimes people will say, I don't know whether it's a supraventricular or a ventricular arrhythmia, and then they're going to give you supraventricular arrhythmia, same patient, okay? Same disease, same thing. You get different diagnoses, and they belong to each other through a hierarchical system. It's very important. That makes analytics enormously simpler if you know that these things belong to each other and in which way they belong to each other. And you don't have to do it in Wikipedia and figure it out yourself. Okay? That is also part of the vocabulary download that you have to take. So yes, you have to download it, and you have to apply it, and you have to load it. And sometimes it's painful, and you get a lot in the forum. You get a lot of those. Oh, it doesn't. How do I load this? But once you've done it, will make your life tremendously easier. All right. Just to summarize the takeaway, when we standardize the format into objects and relationships. Remember that, with the fields, and the content, the reference tables, the value sets, the vocabularies, in order to be able to do tools and data exchange. If we don't do it, we actually can't do much. And the data need to be in a closed world assumption for the tools to work, and they have to be normalized, because otherwise, again, you have the same problem. You have to then do these things on the fly at analysis time, which is sometimes almost impossible. And the vocabularies, we need to represent all facts. It's a closed world. We need to know everything that could happen. And we need to categorize these facts and organize them into hierarchies. And that's what you're provided by the OHDSI vocabularies. Okay? Any questions? John.
Are there types of use cases that OMOP isn't good for delivering?
Are there any types of use cases that OMOP isn't good to deliver? Yeah, all the ones which we haven't actually introduced yet. We don't know the answer to that. But usually when people say, I want to do patient care off of OMOP data, then we say, you don't want, we haven't. This is not optimized for that purpose. You lack transactions, you lack orders, you lack schedules, you lack requests, you lack refusals. Patient, had enough and went home. So you don't have that. You don't have any of the personal information. Patient was referred to Dr. Jones, not in OMOP. There is no Dr. Jones in OMOP. There is a provider, 12345, with a specialty of pediatrician, but we don't know the name, and we don't want to know the name. And we can't know the name because the name would have to be part of the vocabularies. But we're not putting any Dr. Joneses in there. So that's essentially what it is. So if you want concrete information about a patient, don't go into OMOP. If you want to figure out populations, like all patients who have, then that's exactly what, you get exactly what you need for that. And that's how Erika said that's how the OMOP CDM came about. There was an OMOP experiment. And the OMOP experiment had to make a huge amount of comparative calculations using different methods. And so the idea was, oh, Jesus Christ, we have all these methods. Let's at least make sure that the input is always the same. That's the birth of the OMOP data model. Okay.
If I need help mapping my data set, for example, like combat data, into for the new registry we're building, the DoD, how do I get help with our unique concepts from combat to be able to put it into this model?
Yeah. So if I have new vocabularies that I need, how do I get it into the vocabulary? in this case, combat information. But this happens all the time. There is a mechanism called community, what's it called? Community service? No, what is it called? Something with community, I'm going to say it. It's on the web, okay? And that's where you do it. You go. So the first thing is you go in and say, in the forum, has anybody done combat data? And then usually somebody raises their hand and said, oh yeah, I've been talking about it. If not, then you already declared that you need. And if you you can then start interacting with the team to add this stuff. There's very little bureaucracy. You have data, you need it, you're saying, okay, standardize it. And if nobody objects, then that's the standard, and it goes in. There's not much of, there's no committees or ballots or anything like that. It's very simple. You good? Thank you. Who's next? Fan is next.
So this part is not really about doing new research on methods, but kind of about the methods that we have spent time researching on and having discovered that they do work very well in practice. And so we have standardized them, made them available so that you guys can all use them when doing your research. So the content of this part will be like this. First, I will give you a very brief overview of the types of research methods that we do have at OHDSI, which are, roughly speaking, 3 parts: characterization, population level estimation, and patient level prediction. So I will go through characterization and then patient level prediction, and then focus a little bit more on population level estimation because that's more relevant to our example study that Erica introduced in the beginning. And finally, I will touch upon kind of the importance and the work that we have been doing in terms of developing distributed learning methods on a federated data network. So a federated data network is exactly the OHDSI data network. So we have databases that are distributed at locations, so we're not pooled together. So that's why this is important. So let's revisit Christian's patient diary schedule table, but in a picture-ish way. So in this little cartoon, I'm kind of trying to represent a patient's journey through time. So from left to right, you have from history to present time. And different things can happen to a patient, including conditions, drugs and procedures that are administered, and measurements that are taken over time. So suppose we focus on a particular diagnosis or a disease that happens right over here. Yes, you can see my cursor. And then later on, a drug is prescribed or administered to this patient. Usually we call this time point the index time or time 0. So we kind of want to know what has happened or what is happening up to this index time point. And we also want to know what happens in the follow-up time after treatment initiation with respect to this drug. So there are different questions that we may want to ask in terms of what happens with the patient or patients that are very similar to this example patient. For example, what kinds of patients have chosen what kind of treatments after getting diagnosed with a certain disease? So in OHDSI lingo, these sorts of questions belong to clinical characterization. And another example question is, we want to ask if one treatment option is more likely to lead to some desirable or undesirable outcome compared to an alternative treatment. So this is basically trying to compare the effectiveness or safety profile between a target drug and a comparator drug. So that kind of question belongs to the category of population level estimation. And finally, we may actually want to do a little bit of prediction, trying to see will a particular patient experience a particular outcome of interest given the diagnosis and given the treatment that we have chosen. Then that belongs to the category of patient-level prediction. So it's a prediction done for a specific patient given what we know about this person. So together, this is kind of like a triad, or the three pillars that Erica mentioned about the methods that we have developed, validated, and implemented efficiently over the years. So clinical characterization, in short, is about learning and characterizing what has happened and what is happening out there. And patient-level prediction is about predicting what will happen to a particular patient. And population-level prediction is more of a causal ask in nature, trying to learn, trying to understand how and or why something happened. So I'm going to first briefly introduce clinical characterization and how we do it. So within OHDSI, we have an established framework of doing characterization studies. So to do this, you will need some key ingredients. So later on, after the break, Martin will walk you through Atlas. I'll walk you through how you define cohorts, how you go through that. So I'm going to do it conceptually over here. So first of all, you will need a target cohort. Basically those that you want to characterize, you want to learn about. Oftentimes you might need to do some stratification, split your target cohort into subgroups, for example, dividing by gender, race, ethnicity, medication history, comorbidities, and so on. And I think also oftentimes you need to have feature cohorts. So the bunch of people, the bunch of time intervals that you extract features, you extract characteristics, or you extract the outcomes from for your characterization study. And also very importantly, you will need to specify the time at risk window. So you need to have the windows of time during which that you run the characterization study. So here I say the word cohort, and I think Christian also mentioned the idea of cohort. So I want to emphasize again that within OHDSI, the way we think about a cohort is not just a bunch of people. It's actually two things. It's the set of people and also the time intervals, the time window. So it's person times as a cohort, not just persons. And later on, through Martin's demo on how you go about this on Atlas, you will notice that you almost always have to think about and specify time, the time window, when you do OHDSI studies. So that's very important. So now here I'm going to very briefly go through a case study that's actually very relevant to our example study. You will notice if you look really hard that the first author is the same person. So he led George Ripsack led a treatment pathway characterization study, which was published in 2016 to characterize treatment pathways for 3 diseases, including hypertension. They used 11 databases spanning 4 countries, covering more than 250 million patients to understand their treatment pathways for those 3 diseases. So specifically, this is a screenshot from one of their figures. This is what they got in terms of drug utilization, the proportions of patients who initiated on different kinds of medication options for treating hypertension. So notably, I think Erica actually already mentioned this, hydrochlorothiazide is the predominant drug as the starting option, but it's often paired with another active ingredient in terms of combo therapy. On the other hand, lisinopril is the more significantly predominant drug option as monotherapy. So you're able to get the sort of information that's really informative about what's happening out there in terms of healthcare practices through characterization studies. And as I've kind of included on the bottom of the page here, we have open source tools, we have both softwares for you to run this, so you don't have to run a whole lot of code to do this. This is a very automated pipeline, easy to use framework. So kind of just to summarize a little, the characterization is about learning what's happening, what has happened. So it gives us the full picture about healthcare practices and patterns and trends throughout space and time. So what's really appealing about running a characterization study across the OHDSI network is that you not only get information about your local databases, but you have access to a global knowledge network that can give you information about the patterns and trends across geographical locations and across time as well. So now I'm going to move on to introduce patient-level prediction. As I mentioned, this is about predicting what will happen to an individual patient. So let's go back to that little cartoon picture of the patient journey and kind of abstract it a little bit. So given the drug or the treatment administered at time zero, the index time, the prediction task is about telling whether or not a certain patient or whether or not a certain set of patients will experience an outcome of interest during a specified time at risk window. So it's really about predicting whether or not something of interest will happen. So if you have learned about machine learning stuff before, so can I do a little bit of survey here? Who has heard of machine learning or who have taken a machine learning course or workshop? Oh, so it's actually a fair proportion of you guys. Yes, so if you have learned about or heard about machine learning, so there's a super popular task within machine learning that's called binary classification or binary prediction. So what we actually do here is basically exactly that. So suppose the patient having the outcome in the time at risk window is labeled as 1. Not having the outcome is labeled as 0. So if you abstract it in that way, we are actually doing binary prediction at the patient level. So we feed in the patient basic characteristics, and we want to output the 1 and 0 labels about whether or not the patient has the outcome. So let's kind of go back and revisit hypertension yet again. So in here, let me use the example of lisinopril, since it seems to be the most predominant monotherapy option. So let's think about what will happen to a new user of lisinopril within 1 year of treatment initiation. And let's try to predict if this new user of lisinopril will have acute myocardial infarction within 1 year. So let's kind of put it into the OHDSI typical template of specifying which is which. So we need to be clear about what is the target cohort. So the target cohort will be lisinopril new users and within 1 year of treatment initiation. And the outcome of interest is acute MI. And the time at risk window is more specifically 1 to 365 days post-treatment initiation on this drug. So basically, once we have that, we have a very large tool stack for learning or fitting or for giving you a prediction model that's that does this kind of job. So you put in the baseline patient-level characteristics inside some kind of computerized model, which we call prediction model. So here, this can be a black box. This can be whatever binary classifier or binary predictor that you can think of. You put those information in with respect to the T, O, and time at risk that we have just specified. And the model can output a prediction on the outcome, whether or not the patient will experience acute MI or not experience acute MI within 1 year. So that's kind of the abstract template, the framework, and the kind of the nature of the problem. And I kind of want to emphasize here that actually a lot of healthcare considerations, a lot of healthcare problems will oftentimes involve doing some sort of prediction. So if you were at the main session yesterday, you would probably remember that one of the Titan Award winners for this year is a team of surgical sciences unit in Denmark. So they won the award because they integrated patient-level prediction techniques within patient care. So that's kind of a piece of real-world evidence that this is actually important and does contribute to how we do patient care. So in terms of how we do this, how we materialize this within OHDSI, so this is getting to the more technical bit of this part. So bear with me for a second. So this is like a diagram that shows different components of the very large but also very principled and automated pipeline at OHDSI to do prediction, to solve prediction problems. So this is split into multiple components. So first, we actually have to curate the data because the data exists in the longitudinal way of patient records. We don't automatically have the labels, the 1 and 0s of whether or not a patient has an outcome in the way that we have specified. So first, we have tools and algorithms to build the data and squeeze them into the right format for us to feed into a prediction model. And the second component, this kind of touches on the classical machine learning principles for those of you who have learned about machine learning before. So it's really important to not use all the data to train the model. It's very important to split the data available into, say, a training set and a testing set. Within OHDSI, we do a, for example, 65% and 25% split. The larger part is the training data. And we do this because ultimately we want to apply the prediction model on data on patients that it has not seen before. So we need to evaluate model performance on data that is not trained on. We need to know if the pony knows new tricks by learning some old tricks that are somewhat similar. So it's important to get very good accuracy, very good performance on testing data that is left out of the training process. And so what exactly is the prediction model that I have talked about? So I have been very vague about what model that we're actually using. So the reality is we use a bunch of models and we make them available through the open source R packages that I'm listing down here. So you can do pretty much whatever is already available in terms of machine learning tools, from very simple classical ones to very fancy, very modern, and nowadays very popular ones, including deep learning models and ensemble learners. The work group, patient-level prediction, have put in tremendous efforts in making them available and making them very efficient across large-scale data sources. And finally, having the prediction model itself is not quite there yet. We have to see how the model is performing. So we have to run assessments, we have to run diagnostics to see if the model is doing a good job. So typically we would check, for example, sensitivity, specificity, false positive rates, false negative rates, and so on. Kind of going back to the prediction problem of whether or not lisipronil new users will experience acute MI within 1 year, I actually, well, not I, but the patient level prediction team led by Jenna Reps, they have provided, they have already done this prediction job, and they have provided a public available results, our Shiny app, and provided diagnostics on this link. So if all of you try to click on this link right now, the server might be really clogged, but you can definitely play around with this later after the tutorial. So I'm not gonna click on it right now, but I'm gonna show you some screenshots of what kind of information is available up there. So for example, you can play around with the decision threshold in the slide bar here and see what kind of model performance is yielded by different choices of decision thresholds. You can also run diagnostics and assessments on the training data, the testing data, and you can also do cross-validation by kind of switching around which portion is the training, which portion is the testing or validation, and look at the ROC plot, for example. So we would typically like to see this bulged shape. This basically means that our machine learning model is doing much better than a random guesser. And I guess for me, at least, the best part is that you can actually look at the coefficients that explains the association of the predictors and the outcome of interest. And you also hit this little button to download the model for your future use. The really good thing about the OHDSI tools that we provide is that you don't need to build this beautiful, nice R-Shiny app yourself. We already have the infrastructure available. We have already code and tools available for you to pretty much automatically generate the R-Shiny app and launch it on a publicly available server so that the world can see your results, the world can look at your models as well. So finally, I'm gonna talk about population level estimation. So as I already introduced, population level estimation is about getting to understand how and why something happens. So if you guys have heard of causal inference before, then this is pretty much doing causal inference using real world data. So can I do another round of show of hands to see if someone has heard about or learned about causal inference? It's a slightly smaller crowd, but yeah, so about a third or a fourth of us. So don't worry if you have no idea what causal inference is. I'm gonna do a very simple, not quite right, but mostly okay, cartoonish explanation of what we are trying to do in terms of causal inference. So let's really touch upon our example study that Erica already introduced. So the question here, or one half of the question that we try to answer in the Ripsick et al. 2020 paper is, does chlorthalidone reduce risks of cardiovascular outcomes more than hydrochlorothiazide? So remember that the second drug is the more popular one. But somehow the first drug seems to be recommended with more benefits, things like that. So we wanted to do an observational study using our databases to run an investigation of that. So if we try to unpack this question and try to fit all of these elements into kind of the OHDSI template of how we think about analytics, so it generally falls into this sort of template question. So we're trying to compare the risks of a certain outcome or a set of outcomes between a target exposure and a comparator exposure during a specified time at risk. So if we kind of write down all the details in this little list, the target exposure is chlorthalidone, the comparator exposure is hydrochlorothiazide, and the outcome is, actually there are more than one outcomes, but one of the outcomes is a composite cardiovascular outcome. So this includes acute MI, hospitalization for heart failure. Apologies, somehow this got condensed into a weird sentence, and also a stroke. And the time at risk, here we, in this paper, we actually tried 2 different definitions of time at risk, but one of the definition is called on treatment. So the risk window would start after treatment initiation and would end at treatment discontinuation when we no longer observe that the patient is still taking the medication. So essentially, if we abstracted one level up again, we're actually trying to extract causal effect estimates from observational data by comparing 2 treatment options and see if one is somewhat better than another. So we're trying to attribute differences in the or ratios of risks of an outcome to differences in treatments and see if there are actually notable or significant enough differences. So what exactly is causal inference? What exactly is causal effect estimation? It's really about asking the question of what if. It's about comparing counterfactual with factual and see if a different choice, a different, say, a treatment option would induce differences down the road in terms of patient outcomes. So suppose we have a bunch of people. In this little picture, I have 3 different people. So let's imagine in like some kind of imaginary universe where we do have access to a time machine. So we first assign them with clothalidone, the red pill right here. We do that, and given time, we observe their outcomes. And then we take them back in time using the time machine and then give them the second drug, hydrochlorothiazide. So what we have done here is some kind of thought experiment when we take those people back using a time machine without changing anything except the treatment, the drug that we put them on. So in this kind of very hypothetical, not really realistic setting, so we are exactly copying those 3 people twice on some kind of alternative universe timeline, keeping everything the same except the treatment. So if we summarize their outcomes, also summarize those 2 parallel universe treatment options, and do a little bit of differences and see what the differences are in terms of their outcomes when we defer the treatment, then that difference reflects some sort of causal effect when we try to compare which drug could be better for the patients. Now, unfortunately, we still don't have a time machine yet. Please tell me if you have already invented one. I have not, at least. So what we can do in real life, sort of the gold standard or the best option for doing real life causal inference is through randomized controlled trials. So we toss a coin and completely randomly assign patients to target and comparator treatment groups. And through randomization, by probability theory, by statistical theory, we know that randomization can balance the baseline characteristics of the 2 cohorts of patients. So then we can think of those subjects in those 2 cohorts as exchangeable people, and causally they are comparable. So if we see population level differences between those 2 groups, given that we can assume they are exchangeable, then the differences do reflect the causal effect. However, so that's the second however here, for observational data, we don't even have the option to do the coin toss and put people in different treatment groups. The data are already there. Treatment assignment already happened. We cannot do the coin toss. So the next best thing we can do, which is also the nowadays causal inference paradigm, is to model and learn about the patterns in treatment assignment using something called the propensity score. So basically, what we can do and what we typically would want to do is to learn the probability of getting the target treatment given a specific patient's baseline characteristics before the time point of treatment assignment. So it's a probability, it's a number between 0 and 1, and this probability is what we call a propensity score. So statistically speaking, the propensity score is this magical one number that can summarize the patient characteristics, which could be tens of thousands of covariates that come out of patient records that represent the overall tendency of this patient to receive the target treatment. The really nice thing about that is it's kind of the proxy of telling us whether or not people or cohorts of people are comparable. So for example, if this green top guy and this yellow top guy, we somehow learn their propensity scores. So the green guy has 0.7, the yellow guy has 0.68, so those two numbers are rather similar. If that is the case, then we can actually assume that the green guy and the yellow guy are very similar. And for a causal inference perspective, they would be quite comparable. So if they end up in different treatment groups and they have somewhat different outcomes, then the difference in the outcome can represent some sort of causal effect. And that extends to the population level as well. So if the distribution of the propensity scores in the target cohort and the distribution of propensity scores of people in the comparator cohort, if those two distributions tend to be similar, I will show a plot later on. If those two things look kind of similar, then it kind of indicates that the two populations and the two cohorts are also quite similar. So we can make a good causal comparison and draw some causal conclusions. So let's kind of move away from the cartoon alternative universe thought experiment and kind of talk about what OHDSI does, what kind of tools OHDSI provides in terms of making causal inference happen when we use observational data. So I think the central mission here is that we don't really have access to a randomized controlled trial. So we want to try to emulate the features of a randomized controlled trial using observational data. So as I have already mentioned, one of the very useful tools is to use propensity scores to model a treatment assignment mechanism. So what we actually have done in OHDSI is that we not only use propensity scores, but we use large-scale propensity scores. So the large scale indicates that we use a very large set of patient covariates, patient baseline characteristics to fit propensity score model. So it's a kitchen sink, data-rich approach that can predict whether or not a patient have received the target treatment really well. So we can compare the propensity score distributions between the target and comparator cohorts and check if those two distributions overlap fairly enough. If they do, then we know that the two cohorts are reasonably comparable. So that's kind of a desired state that we call empirical equipoise. At the same time, we have also developed strategies for using propensity score to adjust for differences in baseline characteristics between the two cohorts. So we have tools and methods for propensity score stratification and matching and weighting as well. So typically we would do and recommend propensity score stratification because it's a more stable strategy that typically generates high quality results given our own experiences. But there's only so much one can do with available information. There would be information that we just simply cannot measure or simply cannot observe. So there usually would be some sort of leftover or residual bias, no matter how hard we try to squeeze out of observed information. So what we additionally do is we do something called empirical calibration. So we calibrate our causal estimates using an empirical approach. So what we do is that we have a data-rich algorithm actually developed by Erica data that mines our knowledge base, mines databases to find control outcomes that we know have no known association with the exposures of interest. And we also have ways of synthesizing positive control outcomes that we know the exact effect size for. So using those empirical approaches, we can actually diagnose and assess the amount of leftover residual bias that we still have after trying existing causal inference strategies. So we use that as a way to calibrate our causal effect estimate to mitigate and reduce residual bias. So here I'm actually providing a screenshot of the actual paper, and in the highlighted bits of text, I'm kind of highlighting different parts of the things that we do that I already mentioned in the last slide. So in this particular study, we use large-scale propensity scores trained using more than 60,000 patient baseline characteristics. So that's a huge set of covariates, a huge set of information. And we use propensity score stratification. And given the stratification, we do stratified modeling for the outcome, specifically using Cox proportional hazards model for survival analysis. And in this paper, we assessed and diagnosed and adjusted for residual bias using negative and positive control outcomes through empirical calibration. And so for all of those things, we have open source R packages developed under the Hades tool stack. So even if you don't really know how to do this yourself, we have already implemented them for you. All you need to do is download the packages from GitHub or Cron, and you can run with it. So this is another screenshot of the paper of their figure 1. So in panel A and panel B, what I'm showing is the preference score distribution. So the preference score is an easy transformation of propensity scores. So it's basically the same kind of information. So here, those plots can tell us whether or not there's enough overlap between the distributions of preference scores of the target and comparator cohorts. So usually we would like to see the majority of fitted preference scores to fall between a reasonable middle range so that we know we are comparing apples with apples, not apples with oranges. And also we can check using panel C's plot to see if the covariates between the two cohorts are balanced enough after we have applied propensity score adjustment strategies as well. So usually we would like to see like a really flat blob of points. So on the x-axis, those are the differences in the covariates before doing anything. So typically those can run really high, but on the y-axis, we are plotting covariant differences after applying propensity score adjustment strategies. In this case, stratification, we would typically like to see that those dots are really low, meaning that differences between the two cohorts are now quite low after adjustment. I think later on, Mark will actually go through more details of study execution and really walk you through the results. So what I'm showing here on the slide is that we have a systematic approach of publicly disseminating and also providing diagnostics, really examining the performance of our methods for studies like this. So actually, this example paper that serves as the backbone of today's tutorial is only one tiny little fraction of a much larger umbrella study that we ran a couple of years ago. So you can click on this our Shiny app to explore. Basically, we had almost all pairwise comparisons between all of the antihypertensive drugs and across a very large, quite large set of effectiveness and safety outcomes. So here I'm highlighting one of the comparisons for hypokalemia that is highlighted in the abstract of the paper. So when you click on each individual result from each individual database, we also provide a whole slew of diagnostics that look at the quality of the evidence we are generating. So what we try to do here is not just about spitting out a number that may or may not be a reliable estimate of the causal effect we're trying to do it in a principled way such that if this piece of evidence is not reliable, we don't even show it. We just blind it, we throw it away. We only show those results that we know have passed our series of diagnostics that we know is reliable enough. So that's when we show it. And when we actually demonstrate it in a publicly available venue, we also provide all of the information for assessing and diagnosing the model for everyone to see. So it's a very open science, very transparent approach that we adopt here. So finally, let's kind of very briefly talk about how to move towards building a really global or international evidence generation network. So healthcare data, they are essentially localized. The patient-level information, those records cannot move outside of individual database locations because of regulations, because of privacy rules. So the traditional way of kind of synthesizing evidence across different databases is to try to find some way to meta-analyze point estimates and confidence intervals. So we rely on highly summarized, highly aggregated information that comes out of individual data sets and do a little bit of statistical modeling, and hopefully we can get the right answer. But for us, for OHDSI, we feel that that's not quite enough yet. We feel like we can do better. We can actually do much better than simply getting out 3 numbers from one database. We think that we can actually scale it up and try to extract infinite dimensional information by extracting something called the likelihood function. So if you still remember your statistics 101 class from years ago, so the likelihood function basically summarizes the data with the relationship of your model parameters in a much more information-rich kind of way. So this is kind of the direction that we are trying to move towards. We're trying to run hopefully a more reliable and a more information-rich system and algorithm to do meta-analysis, to synthesize evidence across localized data sources. And in a unified framework through Bayesian statistics and modernized statistical tools, we can actually adjust for residual bias in together with doing meta-analysis. So what we can do is we can diagnose biases at different levels, and we can also get a unified answer that we know is bias adjusted already. And we actually already have a package for doing that, and we are actively still developing it to make it better and more scalable, more stable. So just to summarize, what we provide in terms of research methods and OHDSI are actually quite rich. So we have this triple set of methods for different purposes for answering different types of questions regarding the patient journey, regarding healthcare. So we have clinical characterization, patient-level prediction, and population-level estimation. For all of those types of methods that you can use, we have provided a whole stack of open source tools that run efficiently and reliably. And as I already mentioned, we also have the infrastructure and algorithms and code for building web apps that can publicly disseminate and communicate your results. And of course, we also have developed analytic tools for synthesizing evidence across a federated data network. We believe that is the way to go moving forward, kind of unifying the evidence in a more principled, a more reliable way so that we can produce better answers. That's it. Thank you.
Hey,
Oh, that's a fantastic question. So the question is, so in this example, the green person has a Princeton score of 0.7, the other guy has 0.68, then they're quite close and similar and comparable. But what if they are still close, but both of their propensity scores are lower, say 0.3 and 0.28? So the propensity score is kind of like a one-directional score. It indicates the probability that the person gets the target treatment. So in this case, chlorothalidone instead of hydrochlorothiazide. So basically it means if this number is low, then that person has a higher tendency of getting this treatment, the comparative treatment. So it kind of works symmetrically. So as long as two people or two groups of people are close enough to each other in terms of the spectrum of propensity scores, then they are close, they are comparable. So it basically doesn't really matter where they sit, it's really about how similar they are. Yeah, thank you for the question. All right. Thank you. Now it's time for a coffee break, I think.
Okay, so we're done a little bit early. So if everybody can come back at 10:15, so that's about 20 minutes from now, we will get started on the second half of our presentation.
Okay, so how's everyone doing? Everyone
Feeling great.
good time? Yeah? Excited about OHDSI? How about Fawn's presentation? That was really good. I wish she was my biostat professor. That was very good. So anyways, I'm going to talk about open source development, and I've been thinking a little bit about how to frame this because I remember being in a very similar situation to you guys. Came in a few years ago, came into a tutorial class, kind of a little confused, like why are we doing an escape room? Why is everyone so into epi and informatics? But some of what really encouraged me were some of the principles that this community stood by. And the reason why I want to start there is because these principles go from how we think about the data and how we think about the software that we use to execute our studies. This standardization and this principled approach is how we can run these very complex experiments across all sorts of different data sources and produce reliable evidence. So I'm going to talk about open source development. This is kind of a very challenging task because there's so much software in OHDSI GitHub, and you have to install things and you have to know how to run R and stuff, and there's a lot. But I just want to scratch the surface and really discuss what the principles are about open source software and why we're so consumed about standards and why we're consumed about being open source in general. Because that's what helps us achieve this mission that OHDSI is set out to do, which is to generate reliable evidence at scale. So this is my agenda. So I'm going to introduce the tools first, talk about why we're open source, and then why we build these standardized analysis pipelines. And then for a little bit, I'm going to discuss how to use one of the OHDSI tools, Atlas in particular, to build a cohort using the RIPSAC study, so that hopefully you guys read. We'll go through that and discuss how to click our way through constructing the cohort in Atlas, and then I'll take questions. By the way, if you have questions, just raise your hand. I'll take it in the middle of the presentation as well. So here are the OHDSI tools. Well, here are a few of them. So let's start with Atlas. Atlas is the flagship tool of this community. It's a point-and-click web application where you can start navigating analytics on OMOP data. So for people that are just starting out in the community, this is the best way to learn how to use OHDSI software and understand our approaches. Now underneath that sits OHDSI. So this is a collection of open source R packages that allow us to do the analytics that run on OMOP data. So if we wanted to do some of the methods that Fawn was talking about, those are all OHDSI packages. So we can do our statistical modeling in there. We can run incidence calculations, extract features. All of these are open source R packages that are available on the OHDSI GitHub. Then there's some other tools, the rabbits. These are used for ETL design. Athena, which is a web application that helps you search for standardized vocabularies. It was mentioned yesterday by the Vocabulary Working Group. And then data quality tools. So like I said, this is just a few of the tools, but there's so much more. Throughout these slides, I have links that will take you to the GitHubs and stuff. But the thing is, we have all these tools that, and we have Atlas. So this is what Atlas looks like. If you're not familiar, you can click on that top link. It will take you to the demo Atlas. So as I said, this helps you navigate your OMOP data. So you can look at your data source, you can build cohort definitions, you can do characterization, like understand what are the features at baseline. These types of principal methods of extracting evidence. And you can do very simple things here. So if you're just starting out, just being able to understand what a population from a cohort is very powerful. So I've shown a lot of these tools, and all of these are free. You don't need a license. You don't have to pay some company a ton of money to have their software. You can just download this off the internet, which is pretty awesome. So you can just set up Atlas for free. You can set up any of the Hades packages for free, and then beginning analyzing your OMOP data. Well, once you've OMOPed your data, of course. So we do this because we want to create reliable evidence. So in the book of OHDSI, we have this attribute matrix of what makes reliable evidence. So some of these features, like being able to repeat the experiment, and if we can get the identical result, like we want to have that, because then we can say there's no like random process that's taking place that's corrupting our data, for instance. We have reproducibility, replicability, generalizability. So all these features improve the reliability of our evidence because we've run the same experiment across multiple data sources that could be representing different settings, different populations. So how do we do this? We have to have tools that can run these complex experiments across all of these data nodes. And it would be impossible to do this with proprietary software. We have to have open source software because we need to have access to these tools and run them at any site. Anyone can use them and anyone can replicate the experiment that we want to run. And this is also part of OHDSI's principles. Our open source, our open science initiative is based off of transparency and producing transparent, reliable evidence. And that replicates on the software as well. So why are we open source? We need accessibility so anyone can have access to the tools that we want to use. Transparency, so nothing to hide. We can audit these tools. We can make sure they're not some weird black box, because black boxes don't work in our field. Reliability, we can trust what the software is doing. So it says it's doing something, and it's actually doing that, and it's tested for doing that. Validity, we're using real methods that have been published in peer-reviewed journals, things that we can trust, like the mechanics of our statistical models or the way that we're extracting this information. And of course, we have collaboration. So a nice thing is we have a community that we can use to help improve the tools that we have. So if they're open source, anyone can look at them, and if people make mistakes all the time, you can post an issue, tell this is a mistake, this is impacting my study, and we'll improve the quality of our tools, report bugs, and make the software better. So this is why we are open source. So I've introduced some of the tools, talked about why we're open source. So why do we build standardized analysis pipelines? So a lot of you are here because you have maybe your data's mapped to OMOP, and one of the big reasons you want to run network studies. So like I said, if you want to run an experiment on a network, you have to send out your code into the wild, hope it runs, and then get this information back. And that is a very, very, very complicated process. So we have to standardize things. We have to have pillars or things that we can count on. So one of those things is the OMOP CDM. We have a standardized representation of the data. So we know that what we're querying is going to be consistent every single time. And then on the other side, we need to have the software. So everything is contained. Everything when we're running this experiment, it's going to do the exact same thing on these different databases. And we have these problems. So we have like different populations that they capture, different data types. We have different IT infrastructures, which is a very challenging problem. And we have different skill levels. So I may be an OHDSI veteran, but it does me no good if only I know how to run the study and then you guys who are just starting don't know how. We have to produce evidence at scale as a community, regardless of the skill level. So the software that we're creating needs to be run by anyone who has an OMOP instance and can provide information. So this is why we standardize our solutions at each data node. So this is kind of what it looks like. We have this network. So that's me in the middle, a US claims database. I'm on Redshift. But when I developed this study, I got to pass this to my buddy over in Germany who has an EHR and in Postgres. So that's hard because Redshift and Postgres are different. There's this different IT infrastructures. But we need to be able to run this standardized solution across this network and get the results back from all these different nodes. So, I mean, you've already seen, I've shown some pictures of some DBMSs, right? So Postgres, SQL, Snowflake. So prior to my professional work, I was in grad school and I would get all these, I would have to run studies for people. Like a resident would come to me with their crazy Excel file that had all these crazy features, and then I would have to run my statistical analysis. Now, that would be like 50 patients, let's say. Now, let's think of if we took that model and scaled it up, and we're working on MarketScan, which has 80 million patients. How could you expect someone to do a chart review on like this flat file? You get a CSV file, it'd be impossible. So the data is so rich that we have to do this and represent this in an informatic model like the OMOP CDM, which is a simple relational database, something that's been around for a very long time and structurally sound. So we need to use SQL and extract all this information, but like I said, we have all these different dialects. So we have Postgres, we have Snowflake, and everything is a little bit different to how we actually how we're writing that query and how we're extracting that information. So we need to be able to ensure that that query is consistent across each site. So we have the OHDSI community has developed these tools to allow a uniform interface to these database platforms and be able to represent the SQL in a consistent manner. And if we combine these things, we can run the same code at every single site. So one of the things that we know is that we know that each database is going to produce some sort of difference in result. We have database heterogeneity, so this paper does a very good job of explaining that database heterogeneity between the different types of population and the different ways that we're gathering the data, whether that's for reimbursement in terms of claims or in terms of like doctor's notes for EHR. So that's going to provide a difference, and that's okay to have some of that variation. But what we don't want is we don't want to run different queries at each site, because that impedes our pillars. So we know we're going to lose one in terms of like the differences in the database. So to hold up our system, we need another one. So we're running consistent queries. If you were to ask someone to run, say, two coders, put them together and say, you query me acute myocardial infarction, and you also query acute myocardial infarction, you're probably going to get different results if they don't communicate, right? So we need to send just a consistent query across this network that's pulling this data that we can use and then run our statistical models and so on. So this is the idea. We're standardizing our analytical pipeline. So we want to use the OHDSI methods library, or HADES, and Atlas that provides standard tools in order to generate this reliable evidence. Sometimes we write some custom code, but we're trying to avoid that, because any custom code that goes around in the system, it could be unreliable, it's not validated. We want to use things that are consistent in order to run this experiment successfully from each data node. All right, so this is going to be more of like quasi-interactive section of this presentation. So this is the paper that we read prior to this talk, the RIPSAC paper, and I'm going to demo how to build a cohort definition in Atlas. So when we build these cohort definitions, we usually want to go through some sort of process. So process and structure, these are good things, like good things to have, even outside of our tools, like how we approach these things from a scientific perspective is important. So the first thing we want to do is we want to abstract the design. So we want to draw this, sketch this out, make sure it's very clear in our head. So before we touch the tools, you want to make this idea, this clinical idea, very clear in your head before you put it onto paper. Then you want to find the concepts in Atlas, build the concept sets, which I'll demonstrate, build the cohort logic in Atlas. So how do we say, what's the index event, any attrition that comes off of that? So where are the inclusion rules, exclusion rules, so on and so forth. Then we want to generate this, so enumerate the patient counts and then review this. So is that number something that's feasible or logical within from the expected population? And then we can run something like cohort diagnostics to evaluate the cohort definition. I'm not going to show cohort diagnostics because that's a little bit too involved, but that's something that you can look into. So let's start by abstracting the cohort. So I've highlighted, hopefully you can see that, it's a little smaller than I thought. So I wanted to abstract this cohort. So I've taken sections from the study design to build the idea that we're trying to construct here. So it says in the paper, we included all patients initiating antihypertensive treatments with chlorothalidone and hydrochlorothiazide. So as Fawn mentioned, what we have is a comparative effectiveness study. So we have a target, which in this case is chlorothalidone, and a comparator, which is hydrochlorothiazide. So if we're trying to create the cohort of the target cohort, we want to focus on chlorothalidone. So when we're doing this new user cohort design, they have to be clean. So they can't have taken anything prior. They're taking chlorothalidone for the first time, and then we're observing whether there's an increased risk that's what we're estimating essentially. So first time user of chlorothalidone, it also says somewhere in here, maybe, here, at least 365 days before treatment initiation. So this is we've seen the person in the healthcare database for at least a year, so we know that there is enough information that they're providing over time. Now we look at the attrition. So they have to have hypertension diagnosis, so that's the reason why they're getting chlorothalidone. And then they can't have any exposure to a known hypertension therapies, so can't have hydrochlorothiazide or any other hypertension drugs prior. So the third one, no exposure to hypertension within 7 days. I'm going to hand wave that and just pretend that says 0 for the sake of the demonstration. But that was something in the paper that they said, so there was a clean window after, 7 days after. But we're going to pretend it's 0. Now the exit. So this is something that's usually forgotten in cohort definitions, and I remember one of the very first OHDSI symposiums that I came to was I always thought about, we just have the index and then we see the patients. But you also have to think about when we stop looking at them. And that's super important when we think about our statistical models. If we have our time at risk is very important. If we're doing an on-treatment design, we only want to be looking at the patients when they're on the treatment. If we're in intention to treat design, we want to look at the patients until the last amount of time that we can see. So the exit event is also really important in terms of our cohort construction. So in this design, we have an on-treatment design. Well, in the study, they did both of them, but the paper in particular shows the results for the on-treatment design. So we want to look at when the person is no longer on chlorothalidone. Now this is an interesting thing that we do. So usually when you have a drug exposure, particularly in claims, you see the date of the prescription, right? So if you want to create a span of time, what you can say is you have the date, and then if I look 30 days and I see another exposure within those 30 days, I can concatenate those two and start to build an era, is what we call it in this community. So we want to see a continuous era that they're taking the drug. And this represents this on treatment time at risk. So we can change this so you could also do say every 60 days we look and see within 60 days do we see another treatment, then we can add that into the era. So this is what it looks like. I do this a lot in my job. I draw out my cohort, make sure I understand it logically and pictorially. So index event, first time exposure of chlorothalidone, at least 365 days of exposure, have to have prior hypertension, no exposure to hypertension therapies afterwards, and this is the exit. So I draw this. I know there's different ways of doing a cohort drawing. I like to do it, draw the cohort as the logic first, and then you can look at the types of analysis you would do post-index and whatnot in a separate drawing, but that's for another time. All right, so let's attempt to move over to Atlas. Okay. Oh, here we go. Spinny wheel. Oh, we have the man. So hopefully this works. It may be that a lot of you are trying to open it at the same time. So this is Atlas, and it loads. There we go. Okay, so here's our cohort definition. So this is what the interface looks like, and I think Atlas's best feature is its cohort building feature. It does, compared to other proprietary softwares out there, it does a really, really good job of conceptualizing the cohort logic and the framework, and it also looks at concepts or cohorts as not just code lists. So you are adding a list of codes at the end of the day, but it's more than that. It's like thinking about how do we construct this definition as like a computational artifact or like a query. So let's take a look at the concept sets first. So how we build concept sets is, as Christian was saying, we have mappings, right? So to look at ingredients, we look at RX norm, which is our standard vocabulary that we use. So if we look up chlorothalidone as an RX norm through the RX norm vocabulary, which it is done here, I did it beforehand. So this is the concept ID. So the ID relative to the database. So this concept code 2409 is the RX norm code, right? So this is standard. And what we have is the hierarchical relationships. So what we don't want to do is spend all day trying to like figure out chlorothalidone like 10 milligrams or I don't even know if that's real, or like chlorothalidone 5 milligrams or something like that. This brand, that would take forever. So what we do is we find the standard concept and then we add all the descendants. So all those vocabulary terms underneath that ingredient will be enumerated within our cohort definition. So this makes it a lot more efficient in terms of like building these cohorts, and you're not having to do like concept set searches all day. We're relying on the vocabulary and making our decision based off the vocabulary. So we can do the same thing for hydrochlorothiazide. This is the RX norm for it, and then include all descendants. Same thing with hypertension. So we have hypertensive disorder. So in conditions domain, SNOMED is our standardized vocabulary. So we look at hypertensive disorder and then use the descendancy to include all descendants of hypertensive disorder. So if we open this, hopefully this will load. So this will include all these different concepts within here. So there's a lot with hypertension. All of these would be counted. So if we see in our table anything with that top concept ID, that would be enumerated, and anything with these IDs as well would also be enumerated within the cohort. And then finally, we have other hypertension drugs. So we can look up, for instance, like use the ATC code or hierarchy to find what drugs, what hypertension drugs are out there, and then we can add those RX norm codes individually and then include all the descendants. Now, something you can also do is like, say I didn't want to use this drug, so I can exclude it. So this way, when I'm building my concept set, it's going to include every other hypertension drug and its descendants except for verapamil. Yeah. Christian's not here, and he would have yelled at me for pronouncing things wrong, but I am a data scientist, so I'm free of that pronunciation. Anyways, so that's how we build our concept sets. So concept set construction is really important because while we are, so as I said, the nice thing about Atlas is that we have more of a cohort building logic taking place here. It's not just a codeless generator. But at the end of the day, we are still pinging these different codes that we can find in our healthcare databases. So coming up with good concept sets is very important. And there's all sorts of open source tools that will help you recommend the correct concepts. So there's this tool called Phoebe, which is a concept set recommender. So if you give it a few concept IDs, then it will tell you these are additional ones that you should probably include as well. So there's all sorts of these free tools, as I said before, that allow you to build very complex things. So now we want to reflect our cohort logic. So before I was saying we had new user cohort chlorothalidone, right? So we want to do for the first time in person history. So if we wanted to add that, we add this as first exposure criteria. So that's an attribute. And then we want to add with continuous observation of at least 365 days. Now, if we wanted to include inclusion rules or inclusion criteria, we can go down here and then add each of these. So once we have the concept set loaded, we can add that into a particular domain. So if we want to look at a condition occurrence of hypertension, we can add that. And now we need to include the logic, like the temporal logic. So if we go back to our PowerPoint slide. Where? Yeah, I'm trying to. I've lost the mouse. Apologies. Yeah, there we go. So if we go back, sorry. So we go back to our structure. So when we look at these things at baseline, everything is relative to the index event. So we want to have this in a certain amount of time where the corresponding to the index event. So if we go back to our thing here, it's saying we have to have at least one occurrence of hypertension starting any time prior to the index event. So that's how we think of these inclusion criteria. And the same thing with all of these. And then now for the cohort exit, we want to have an end of continuous drug exposure, so we can toggle this, so we can either do end of continuous observation. So if we wanted to do an intent to treat design, we can look at all the remaining patient time that's in the database. But since we're in an on-treatment design, we're going to look at end of continuous drug exposure, so we're going to look at chlorothalidone within 30-day persistence windows. Okay. Now I have to go all the way back down. Silly. Quick review. Sure, I'm giving you all a headache right now. All right. So this is how, so as I showed in Atlas, we build this concept set, we define the logic, and then we can generate these counts quite quickly with Atlas. So going back to the standardization. So what's taking place under the hood is something that and this is a little bit technical, but what I'm trying to show here is we're creating standardized queries. So if we use the Atlas tools and we create this logic through the web application, what's going to come out is this standardized SQL. And that standardized SQL is what we can pass across our network and enumerate this population consistently at each data node. And the mechanism that's taking place there is through this SIRCB engine. So we have the Atlas inputs, they fit into an object model that's JSON, then that gets serialized into a standard SQL, and then we generate and we enumerate an n by 4 table. So that table is what we target for all of our analytics. And that table will include the definition ID, the person ID, the start date, and the end date. So the start and end date is really important because that's how we can determine whether something has happened during a certain period of time. Usually we'll look at that relative to the start date, the index start or the cohort start date. But when we're looking at things like end of events, the end date is also important. So now we have all the necessary features to run the remaining analytics. So how are we doing on time? Okay. Any questions so far? Questions? No. Okay, so next thing is to prepare a cohort method. So when we're doing when we're running cohort method, so we go back to our study, and this is a little bit small, I apologize, in OHDSI, what we like to do is like the kitchen sink approach. So this is large scale propensity scores, as Fawn mentioned. So we're going to take as many features as possible. So we have this tool called feature extraction. It's optimized to look at each domain within our database and extract temporal features. So whether something has taken place 365 days before, 183 days before, 30 days before, and we use those bits of information to build our statistical model. So these are the covariates that we extract. Then we have to determine what is our outcome definition. So there's a few of them in here, but I'm going to use the composite cardiovascular disease outcome. So this is a composite of acute myocardial infarction, heart failure, and stroke. And then we have to think about the time at risk. So since we're a non-treatment design, we start from the day after treatment till the end of the treatment. So that's why the importance of the exit is there. So we know that, that we're going to stop looking, or we're going to stop considering the patient at risk once they stop taking the drug. Now our model is a Cox proportional hazard model, and we're using propensity score strata. So if we go back into Atlas, we'll try this one more time. Okay, so this is what it looks like to build the patient level estimate or population level estimation. So we start, once we've defined all of our cohorts, so we have our target cohort, our comparator cohort, we also design our outcome cohort. Now we can plug these in and start by building our comparisons. So we add our comparison of chlorothalidone as our target, compared to hydrochlorothiazide as our comparator, and then look at composite cardiovascular disease as our outcome. Now we want to look at our analysis settings. So if we look at our analysis settings, we plug in all the bits of information that we want relative to the study. So this is taking a look at a calendar time between 2001 to 2018, right? We want to have at least 365 days of minimal observation. What else is in here? We want to remove subjects that have had the outcome prior to the risk window. So it has to be clean. They can't have composite CV prior. And then we are going to add our covariates. So one thing that we want to do when we build these models is to avoid correlation between the treatment and the covariates that we're considering. So we want to remove any hypertension therapy ingredients because there is going to be a correlation because there's similar concepts of hydrochlorothiazide and chlorothalidone. There's similarities between those, so we want to exclude those covariates from being used in the model. Otherwise, we're going to have like a multicollinearity type of issue. So then we define our time at risk. So this is 1 day, and then from the cohort end date, 0 days, and have at least one day at risk. And this is how we determine our propensity score adjustment. So a lot of this is we're filling out a menu here. Like that there's a reason for this. We're standardizing this design because we want to be able to run this very complex model across several data nodes. So this drop-down menu, essentially what it will do is it will export an R package, and then that R package can be sent out to each of our data nodes and then run, and then they will get like a zip file and then return that to the study host. And that's where we can start to build like our Shiny app to visualize all that data. So that was just a brief tutorial, and like I said, there is so much to introduce in terms of OHDSI tools, and there's so much to explore. But like, what my goal here was to try to introduce the principles of why we have standard tools, why they're open source, and that helps us generate reliable evidence. And that's the most important thing, is that the evidence that we generate is reliable, because we have a reproducibility crisis, particularly in observational studies. So a lot of people can generate random stuff by running a single analytic on a single database. And there's a lot of noise out there. And what we're trying to do is improve the quality of the evidence that we generate and lessen the noise, so clinicians can improve the decision-making process, improve healthcare for people. So that's all I got. Any questions? Yeah.
Can you talk a little bit about using Atlas for this process versus like some of the R packages, the R tool stack, and like sort of the pros and cons?
Yeah, okay, that's a good question. So the question was using Atlas as like your navigator compared to R, doing the whole process in R. So as I mentioned, all the Hades tools are, they're the underlying elements here. And so if I export this, this creates an R package. So what if you were to do the entire thing in R? The advantage of doing it in R is that you have more control. Atlas is a more contained environment. So you have, because it's a point and click application, so there's only so much that Atlas can do. Doing the analytics in R, you have more flexibility to do more analyses, add more things to your study to your study code. So when you're running more complex things, you can do that in R. But I would suggest that most people do, especially when you're running a first-time study, to do it in Atlas and understand the mechanics first, and then you can expand out to more complex studies, and yeah, as such. Yeah, hopefully that answers your question. Any other questions? No. All right. By the way, I have a ton of additional resources. So this has just like several links and bits of information on like how to set up the R environment, which is can be challenging. And my one last thing is recommend just be patient when you set these things up. They can be challenging. Like I said, all these tools, they're standardized, but they're still complex. Ask for help, post on the forums, post issues on the GitHub. That's what we're here for as a community, to collaborate and help you guys. But hopefully some of these resources can help you on your journey. So thanks.
Better grab it. I can't use a Windows system.
Okay.
Yesterday.
Yeah. Should. From here. Okay, there you go.
All right. So I know this is a very large room and it's a little cold in here, and so we're all kind of snuggled in with our hoodies over our heads and our arms crossed. But I'd like to make this session a little more interactive. And one thing to keep in mind is, the tutorial is really put together to not only introduce you through the community and the material that we've put together, but provide an opportunity to find out what are the questions and the on-road adventures that you as the audience and the participants in this tutorial really want to know about. So I'm going to encourage you at any moment to stop me. And if Mui were here, that would be in the form of heckling, which is totally fine. Maybe Erica could step up the plate for that. But also additional questions that you have. I'm going to turn away for a moment from the underlying tool stack that empowers our community to talk at a little bit of a higher level on what we've accomplished up to now with this tool stack and what the steps are that one needs to engage yourself in using it to generate really reliable evidence. And I don't know exactly how this figure was updated in the This is OHDSI report that Craig put together this year. But my most recent snapshot of our community dashboard, which tracks publications from individuals within the community using the OMOP CDM or the OHDSI tool stack, has us at a little over 600 publications across the community. And I've heard in confidential discussions, so I'm going to disclose it publicly to all of you, that this really is the number to beat if you happen to be in one of those other unnameless communities where they're like, we need to start targeting, and 600 is sort of the number that we have to go. So as a community, I think we've been very successful in terms of academic publications. Now, that's not the end all. I mean, I'm an academic, so that's the end all and be all. But I understand that not everyone, well, I don't understand why not everyone wants to be an academic, but I do understand that not everyone is, and there are other priorities besides just publications. But let's just take this as a rubric for the moment. Now, if we break down the topics of these publications, the vast majority of them are focused on medical informatics and informatics building types of audiences. And honestly, I'm a recovering physician and a statistician and don't care anything about informatics. So I look at 600, well, I like to use informaticists, which is why I come and I grab them from the community, but it's not really my shtick. Right? My shtick is how can we really deliver reliable clinical evidence so that we can achieve our mission of providing the evidence that allows patients and providers to make the best healthcare decisions for themselves and for individuals for whom they are caring. Now, in spite of that rather negative statement that the vast majority of that 600 are things that aren't of interest to me, we have had a small number of very high impact examples of delivering reliable clinical evidence. So I'm going to highlight some of those past successes as motivation for us newcomers into the community as directions in which we might want to push this, what tools are out there, and how you might want to get engaged and involved and make these tools better for really delivering more reliable clinical evidence. Fan highlighted sort of the three different pillars that one might construct to describe the different types of clinical problems that we have been addressing up to now. The first of those is related to clinical characterization, and I'd like to remind you of this paper that George led now almost a decade ago, using cohort characterization for the first time at scale across the globe to ask a question about what are the different treatments that people are using when they are first diagnosed, in this particular case, with hypertension, depression, or diabetes. And it was a large success of the community coming together to aggregate information from 11 different data sources, as Fan introduced earlier, covering 250 million patients. And as a success to George and the community, and as an academic, I'd say this is good. It's like 300 citations, and that's a good impactful point at the 10-year mark. Other more recent examples of delivering reliable and useful clinical evidence in terms of characterization come back just a couple of years. The first is looking at almost 5 million individuals who have been newly diagnosed with COVID and asking what are their different comorbidities to be the first global characterization of the SARS-CoV-2 infected populations. And perhaps more important for clinical practice is a paper led by Shintong Li and our colleagues at Oxford in the BMJ, which is the real first to characterize background incidence rates of suspected adverse events after events that are expected to maybe occur or be associated with COVID vaccination. One of the first steps that you might want to ask is, if I want to look for an increased association, I have to know what the rates of these events like Guillain-Barré were before the introduction of the COVID vaccination. And so we've been very good at generating these types of statistics which have an impact on clinical care or our clinical choices. I'm not going to talk about the impact that we've had in terms of patient-level prediction because I'm certainly not an expert in the predictive field, but I would suggest that you reach out to individuals like Peter Reinbeck and Jenna Reps to get some background on where prediction models have actually been impacting clinical care. Instead, I'll step over to the sort of the third pillar, which is looking at causal relationships, and talk a little bit about some impact that we've had in terms of comparative effectiveness and safety. And this will immediately feed us into the general theme that's been moving throughout yesterday and today as a backbone of sort of hypertension and large scale studies looking at hypertension treatments. We published just a couple of years ago the first global study that systematically looked at all 56 licensed agents for individuals who have been newly diagnosed with hypertension in a comparative effectiveness across all of these different agents and simultaneously an evaluation of their relative safety measures. Looking at those safety measures gave us a total of about 32 different outcomes. And although I've only given the masthead here for one of these papers, getting involved in these types of network studies is not a simple, we're going to do one paper and we're going to move on, because we want to explore the richness of what we can learn across the network to provide the best reliable evidence. So this is just one of multiple publications. And the publication that I want to move to out of this large-scale evidence generation and evaluation across a network of databases framework is that which I had to twist George's arm to write, or the Hripsack et al. Paper that you were all assigned as homework. But we'll get back to a quick poll on who actually did their homework kind of later. Before we get there, I want to talk a little bit about the philosophical underpinnings of why you might trust evidence that is generated at large scale and evaluated across multiple populations more so than the traditional observational study. So, legend and its its several incarnations over that time really follow OHDSI's mission in aiming to generate reliable evidence, and in this case, particularly on the effects of medical interventions, different drug treatments, although it can certainly be expanded into uses of different devices or biologics, or any comparative treatments that we have using observational healthcare data. And in order to do this, we did not start off with a specific study design or clinical question in mind, but instead began by asking ourselves, what are the general principles and guidance that we'd like to see followed in any question, such that at the end we can feel confident in saying this is reliable evidence that we'd like to use to change healthcare. And as all good principled type of documents should have, there we whittled it down to 10. You might imagine Martine and myself walking down from the mountain with 15, and we dropped 5 of them. But we ended up with 10, and like the 10 Commandments themselves, they're not necessarily they're very regionalized. But a couple of these principles are perhaps more important and more universal across the whole world than others. And as time changes, our relative value weights on which of the commandments we think are most important changes as well. So, out of these 10, there are really sort of 3 ideas that I think might be a little more universal. And sort of chief amongst these are the idea that one should not be looking at a single treatment comparator outcome by itself, oblivious to what's going on in the rest of healthcare and the community. Instead, one should really try to use standardized tools that aren't shortcuts, but yet a standardization that allow you to develop a complete picture of what a complete picture of what possible outcomes might look like. And the reason why you might want to generate this at scale using not only the outcomes that you specifically are focusing on, but a large number of negative control experiments is so that you can fill in sort of that missing publication bias that we have if you just look at published results. Right? If you just look at the published literature, at least in the observational world, one might argue that there's a huge amount of p-hacking going on. Right? One actually decides ahead of time what is the analysis I want to look at, what is the result I expect to find. I'm not going to pre-specify my protocol because you can only do that in randomized work where they require you to. Why would I do that anywhere else? And then you fall into the situation like this comic highlights, which basically says, you know, I didn't quite get the result that I wanted, but maybe I'm thinking about the problem wrong. So instead of having pre-specified it and run across all possibilities ahead of time, I'll just tweak it a little bit until I end up with the result that I want. Instead, you might include a large number of negative control outcomes to fill in all of those the information about the studies that you didn't run to allow you to adjust for the residual systematic error that might be remaining in the system. At large scale means using the systematic tools that Christian and Martijn and Fan went over. But it's important to remember that systematic doesn't mean taking shortcuts. Systematic means working hard to systematize best practices. And best practices here might mean you need to pre-specify everything that you're doing so you can avoid p-hacking. Best practices in terms of comparative and effectiveness might be thinking about what is the randomized controlled trial that I would like to run if it were feasible, and how can I best model or emulate that trial in the observational data itself. And then adhere to open science or disseminate all of the results that you have, not just the collected ones that look very interesting, but all of the null results as well, which allows individuals who might be interested in a slightly different question to do the appropriate adjustment for multiple comparisons, which you can't do from the literature itself. So George put together a very nice diagram that helps to pull together each of the steps that you might want to consider in delivering reliable evidence that is both verifiable and open. And I'll give you a moment or two to look at all of the different pieces that come into this. You know, we're only going to use validated methods for best practices. They really are going to be best practices, and we're going to accept the fact that there is residual systematic error. And so we're going to try to systematically adjust or control for that bias as well through negative control experiments. Right? Although we're executing at large scale, we appreciate that not every comparison has the data to give us a reliable evidence. So we're going to use extensive diagnostics to rule out trials or experiments for which those results we would not consider to be reliable. In generating many hypotheses at once, that actually allows us to look at what are the operating characteristics of this machine. We're not just going to assume that there's no residual systematic error and nominal operating characteristics are, type 1 error of 5%. 1 out of 20 is, a false positive that I really shouldn't be matching, right? In truth, that's not what happens in the observational setting. But at scale, we can get an estimate of what those real operating characteristics are. And one way to do that is standardization across many different databases with different residual systematic error across different populations, locations, and practice types. In order to make this reproducible and repeatable, as Martijn described, everything's going to be open, from the fully pre-specified public protocol all the way down to each of the different result artifacts, whether or not they make it into scientific publications, reports to regulators, or engagement with the clinical communities. And bringing together, this sort of leads to the 3 R's of of well-done observational research, which is rich in the information that it provides. It's not p-hacked and singly focused. It's rigorous, using systematically done best practices. And because of this and its large bits of, and its incorporation of diagnostics and everything that we're looking at, can be considered reliable with really known operating characteristics. So that's a really tall order that I twisted George's arm and said, here's a specific comparison within this, legend hypertension study. We need to deliver part of this evidence because I think we have something very important to say in terms of clinical care. Now, the next step that George had to take is often completely invisible. And I want to highlight it particularly to this introductory tutorial so people realize that there is a lot of magic going on behind the scenes, but it isn't magic, but it's bits that you need to be aware of to make sure that you're able to get there. And if you're going to execute something that's systematic across a large number of data sources and many different questions with the appropriate negative controls and sensitivity analyses, there's a whole lot of pre-specification and organization that goes into dealing with this type of information. Right? Not only are the raw patient records intractably large to fit in this single Excel file that Martin is really glad that he no longer get dumped on his desk, but have to exist in some sort of relational structure. But all of these study artifacts in this type of reliable evidence is going to have that type of infrastructure as well. And so there's a lot of organization that you need to both use and make accessible and open, not only to your community of collaborators, which are going to exist across many different time zones and several different dominant spoken languages, and different coding practices and regulatory requirements, but also open to the large scientific community itself after the fact. So others can look at the reproducibility and validity of these types of results. I think as a community, as I mentioned yesterday, one place where we probably have failed to yet standardize this type of action is not in the execution of the studies, but in this organization of making sure that the community, OHDSI, the larger scientific community, and the public at all can kind of access these things. And so I want to task each of the relative newcomers here to think about how you could start contributing to the standardization of this dissemination of evidence. One example of the approach is I've put up a bit.ly page to a discussion of a data model, which is sort of the first description of all of the artifacts that come out of using Strategist. And they are enormous. They are so large that they themselves are held on a database server so that they're publicly accessible and usable. We as a community have tools that can allow you to directly interact with these artifacts. And so in the beginning of this document is like, how could I possibly access these if I'm interested in writing a paper? And they build on our Hades tool stack using, for example, here, database connector to get you into the results artifact. And each of the study artifacts, has a table of results. And we are just now starting to standardize what each of those tables should look like. So it's, up to now, what this data model is actually hidden completely inside the code and not really accessible to the clinicians that I work with who want to pull out some incidence rates for specific diseases. And so we need to do a better job. And I think all of us here can engage in doing a better job in kind of describing what these tables look like. And this is just a beginning snapshot of the different sort of data models that we're currently forming for characterization, cohort diagnostics, generation, incidence, cohort method, all the way through. And you might notice someplace where we all can start contributing is this data model doesn't even have a human readable description of what each of those columns are. So there's a lot of work that we can contribute there. But is it really worth it? Yes, it certainly is. Standardization, not simplification, but standardization of best practices, of tools that allow us to take the results of network studies and deliver reliable evidence or summarize this evidence are what we need. And they're kind of under the general idea of if we write them once well, we can use them many. And I invite all of you to join in figuring out how we can write them once well. I've started on some documentation of some reusable authoring tools that allow you to take the results artifacts from network studies and turn them into interpretable scientific publications. All of this now has sort of been handcrafted R. But again, it's R that uses the OHDSI tool stack. Here I'm just using a combination of database connector and some data manipulation, particularly sort of SQL renderer and all the tools within Hades to render, in this case, like a figure that you might see in a paper. And this is the code that we had to put together to sort of render this figure that we ended up using in the SOS challenge. So a good first step are these beginning tutorials that I've linked to here on how you access the results from a network study, and from that generate the tables and figures that help you tell the important clinical study, the story, to deliver this evidence. So let's use some of those tools and look at the artifacts that were generated, particularly at the comparison for chlorthalidone and hydrochlorothiazide. And George was kind enough to introduce this problem in our opening keynote yesterday, and we've touched upon it already in this tutorial, but I'd like to provide a little more background information that will help us interpret the results slightly better. So in cardiology, at least in the US, if you don't want to be sued, standard practice is following the guidelines set forth by the American College of Cardiology and the American Heart Association. These guidelines were updated significantly in 2017 when, for the first time, the guidelines included some prescriptive information on what might be the best agent for a person to start on if they're newly diagnosed with hypertension. And they made a large number of recommendations, which we've looked through in terms of the OHDSI hypertension study. But today we're going to focus particularly on a choice within the thiazide or thiazide-like diuretics. If I'm newly diagnosed with hypertension, should I, for example, start with chlorthalidone, or should I start with hydrochlorothiazide? And kind of the comment in the guidelines are chlorthalidone's preferred because something about its kinetics, it has a longer half-life in people, and what they wrote as proven trial reduction for cardiovascular disease. Now, are there any hypertension guideline writers in the audience before I get really disparaging? No. That kind of misses the point that there's also proven reduction that's versus placebo, and there's actually not a study for hydrochlorothiazide versus placebo. So proven reduction of what? No, there's comparators of kind of different ones. And just because it has a longer half-life doesn't necessarily mean it's a better or safer drug. So I said, George, we really got to look into this. This is kind of a debatable recommendation. And one of the reasons particularly why I thought it was really debatable is I've not practiced medicine in 17 years, but I've written exactly 0 prescriptions for chlorthalidone. I trained on the West Coast. I had never even heard of chlorthalidone. As far as I understand it, this is totally a regional thing. Like, I talked to my colleagues in Europe and they're like, what drug? Like, it's just New England. Like, that's like a New England thing. But like, Who's writing the guidelines? Okay, fair enough. So there's some regional preferences. There's some in vitro evidence that it has a longer half-life, but there is no, or had been, no head-to-head comparison of these two drugs. So it's, maybe it's a good idea, maybe it's not. There was a $10 million investment through the VA, and our colleagues here from Vinci were actually in charge of the analytics for this randomized controlled trial. It's arguably the largest and maybe the last randomized controlled trial that will be done for first-line agents for hypertension. And it was an amazing amount of work. But even it was not focusing on newly diagnosed individuals with hypertension. It was taking individuals who were on hydrochlorothiazide and then randomizing half of them to take chlorthalidone. So not quite even answering the question that's here. But at this time, it wasn't due to report out until 2022, and this was 2017. So there was a homework assignment. The homework assignment was to read this paper. Anyone want to admit to having done the homework? Not done the homework. Anyone admit to having done it? Yeah, there was homework. All right, I like that admission. Okay, fair enough. Well, this is going to be a very interesting discussion, isn't it? So I twisted George's arm, and we published the results of our comparison in this JAMA Internal Medicine paper, which we're all reading right now, or at least you have it up on your screens. And the important thing to remember is this was published like 2 years before the VA DCT randomized controlled trial was scheduled to report out. And in looking through this observational study, some things that we need to do in terms of proving or demonstrating kind of the reliability of the evidence is asking, if you read through it, do you believe it's rigorous and is it reliable? And so some of the things that you might want to initially look for or report out if you're writing these things are what are some of the strengths that you're coming in with, and what are some of the largest weaknesses? Well, I think George did a very good job of highlighting some of the strengths right in this design setting and participants. So in this case, you don't even have to get much further than the abstract. And one of the big strengths here is, although it's an observational comparative study, there was both large-scale propensity score modeling, which I talked about yesterday, for handling not only measured confounding, but indirectly measured or unmeasured confounding. And at least for a trial of hypertension, that's pretty important. Most administrative claims data do not have blood pressure measurements, but they do have a wealth of other measurements that are highly associated with blood pressure. And so if you can do a large-scale controlling for what you do observe, you're probably doing a good job of the adjustment for blood pressure itself, which would be a major, major weakness of this type of study. In addition, there's a large number of negative control experiments because things have been standardized and you can re-execute across a large number of different outcomes, which give us the ability to quantify the residual systematic error. Is there residual systematic error or bias in this observational study or not? And quantify how reliable are our suggestions of significant results, knowing the operating characteristics. The next thing you might want to look at or report if you're describing a study like this is what exactly are the patient populations for whom you're reporting. It's important to remember that randomized controlled trials are executed on very limited, very homogeneous populations, which are not necessarily representative of all new users of some drugs out there. So this particular study, the study populations that passed rigorous diagnostics were in the US, so this is valid for US patients, from 2 large administrative claims databases and a large aggregated electronic health record covering almost, 750,000 new users of either of these 2 drugs across the US. You're really limited with the amount of patient characteristics that you can provide in the paper itself due to sort of size limitations, and this is just kind of a snapshot of the beginning of the baseline characteristics of the different groups, so you can ask yourself before or after adjustment for differences in treatment assignments, are these comparable populations, and have we adjusted for any confounders that might be associated with the assignment to the different treatment and the outcome itself? But we can do much better because we have an extraordinarily rich characterization of these populations. And so one thing that I highly recommend when you start putting these types of documents together is to deliver that rich information itself. A place that I'd suggest that you go, but I'm not going to because my skills on a Windows computer don't allow me to jump back and forth between windows, is the Legend Basic Viewer, where you can pull up not only the study artifacts, but a detailed description of each of the different populations. So you can find what you think might be your most favorite confounder and ask, is there appropriate adjustment before and for these different characteristics? And some important confounders here would be sort of the disease, the stage of hypertension, and other comorbidities of the patients that are sort of hypertension or at least syndrome X sort of related, so hyperlipidemia, diabetes, etc. I would argue that we have the ability to provide all of this information, and it should be reported for all cohorts and all of those that ended up being meta-analyzed, so which passed the rigorous diagnostics and meta-analyzed. And the reason I would argue that you provide all of it is there are important questions. Why did a specific set of individuals not pass rigorous diagnostics? What is it about that population that's different than the other databases? And knowing about those differences gives you information about how generalizable are the results that you're presenting. As Fan introduced, to adjust for both measured and indirectly measured covariates, this paper used large-scale propensity score modeling. And in this approach, we are making a prediction of the treatment assignment process that's going on in the patient and the clinician's mind when they decide which of the treatment should an individual start with. And at large scale, because we have rich standardized approaches, we can include, in this case, between 60 to 70,000 different patient features. There are a number of diagnostics that we've described that you should be very certain to include in the types of publications that you're putting together that will allow readers to just to judge how well were you able to adjust for this type of confounding. And why would you do this? Well, there is really a very strong belief. I would say it's almost surely for the statisticians amongst you that in an observational study, there will be a strong association between either something you measured or you did not measure that you need to adjust for in the study that you're working at. Now, it might seem a little incredible that you can do a predictive model on individuals with 60 to 70,000 covariates across millions of individuals, but it's not really. It's just a little trick through statistics using some regularization, right? So we allow the data to tell us which of those 60 to 70,000 features are predictive of that treatment assignment and do that model selection simultaneously. Out of building of this model itself forms the basis of several of the very important study diagnostics that Fan went over before, including both empirical clinical equipoise. Is there a belief that these two treatment and comparator cohorts actually could have been randomly assigned in some randomized controlled trial, or is there no comparability between these patients? And additional information about how comparable are these cohorts with respect to each of their measured covariates through the covariate balance itself. We are an open science community. We have developed a large number of study artifacts. I would say it's important as a community that we work on delivering all of these. And you will find if you go to the Legend Basic Viewer, anyone have it opened up? Nice. Okay. And so you can actually look at the study artifacts specifically for chlorothalidone versus hydrochlorothiazide for any outcome of your particular interest, but you can focus on MACE, called, I don't know why I called it cardiovascular disease. Well, it's not. Yeah, please.
Sir, on the slide, what is covariate, is it talking about hypertension or like chlorothalidone or hydrochlorothiazide, or is it mean something different? What does covariate mean?
Oh, it's a very important question. I don't have, so I have a good answer to the question. I don't have a good name for what to describe this. The way you will hear people talk about the patient features. So everything that we've learned about individuals in the target and the comparator cohort up to the point of pseudo-randomization at the index date. So if I were writing a publication, I would say these are the baseline patient characteristics or features. And to make that a little more concrete, age, other comorbidities. In practice, there's a standardized tool called feature extraction, which generates these covariates or features or baseline characteristics, and you have a good control over how you want them constructed. You have a complete EHR, so this might include every single condition occurrence code, every single drug, they're called drug occurrences. I don't know. Anyway, it's something in the drug table, right? At multiple different time points before that point of pseudo-randomization. Obviously, if you use information after that point of pseudo-randomization, then there's some causality problems that you have to deal with. But we're building a predictive model. Should they have, which treatment did they get, and with which, with which predicted probability? And it's totally fair game to use anything you know about the patients up to the point when that happened. Does that help answer the question? I'm thinking no, because you're still looking like, what is he talking
I
about?
kind of understood that. So covariate is like age or sex or medication.
Yes.
Or
Yeah.
like in this case, chlorothalidone, it's some variable.
Well, it's some variable, but in this case, it's actually not chlorothalidone or hydrochlorothiazide. This is, here, let's go back. So here's an example. So amongst the individuals who are new users of chlorothalidone, here's a description of their age feature, covariate or baseline characteristic. This is the proportion that are female. And you can imagine this list is now going to go down 60,000 more rows. So, do they have a diagnosis of diabetes? Have they had a diagnosis of major depression between 1 year and 5 years before this time point? Does that help? Yeah, okay. Perfect. And there's a lot in sort of the study artifacts that are not initially going to make it into the publications that you're writing yourself. Here I'm bringing up sort of talking about covariate balance. So this is how similar are individuals who are newly treated versus with chlorothalidone versus hydrochlorothiazide after I've adjusted for their probability of ending up in each of the different cohorts. And I've clicked on the meta-analytic result, so it actually shows you across each of the different databases how those things differ. And a take-home message that we can talk about later is, you want this number to be very small after you've adjusted for all of those differences that you've been able to measure, and very small is generally taken to be like less than absolute value of 0.1. Obviously, before adjustment, there are natural differences between patients on chlorothalidone and hydrochlorothiazide. Like everyone with chlorothalidone was treated in New England, and everyone treated with hydrochlorothiazide was treated elsewhere. Those types of things. Not only are we going to run this experiment for our outcome of interest, but we also need to report the results that we got from all of our negative control experiments. And the negative control experiments allow you to measure as a diagnostic, for example, the ease, and then later adjust for the residual systematic error that's inherent in any observational study. Particularly in legend hypertension, we used a set of about 75 different negative controls. And the design, best practice design that we set up ahead of time, even without adjusting for the residual systematic error, showed very little bias. So it's a good head-to-head comparison in this case. You might keep in mind, and you might argue in a number of your papers, that the diagnostics that you get from negative controls can also allow you to compare different design choices. In many cases, there are several different sensitivity analyses, and there is lack of consensus on what is the best design to reduce residual systematic error. Well, you can measure residual systematic error as a pre-step before you specify your whole study to pick a design that has the smallest residual systematic error to begin with. Let's briefly, given the time, look at the major outcomes for this study. You can, report characteristics of the individuals, so those starting on chlorthalidone versus hydrochlorothiazide. We have the 40,000 people from New England versus the 700,000 people from kind of the rest of the country. And, some hazard ratios. It's probably easiest to look at the calibrated for residual systematic error hazard ratios across the 3 different data sets and a meta-analytic estimate, which strongly support no increased risk or no difference, or strongly do not reject a difference in risk for major cardiovascular outcomes, irrespective of which drug you looked at. But I said you should do things at scale. And it's great to report that there's no difference in terms of effectiveness, but maybe there are some differences in these drugs. And so best practices also, given a large number of outcomes, allow us to look at safety outcomes as well. And this is a hard to read forest plot from the paper itself, where we're looking at the estimated hazard ratio, comparing the 2 different drugs across a large number of known safety side effects for different hypertensive agents. And although there's no difference in cardiovascular effectiveness between these drugs, it does appear there is a quite large difference in these drugs when it comes to their renal function. And in particular, the more potent, which was assumed to be a good property of chlorthalidone, actually leads to significant higher rates of kidney dysfunction, and in particular hypokalemia, and that's to be expected for a loop diuretic, particularly a very potent loop diuretic. We are also, because we're doing things at large scale, able to incorporate a number of different sensitivity analyses. One important sensitivity analysis for hypertension treatment for hypertension might be including blood pressure measurements. And in a number of the EHRs, we do have blood pressure measurements. And so you might ask yourself, if in data sources where I have blood pressure, and I use that baseline blood pressure as an additional covariate with which to do my adjustment and balance, does that change my results very much? And in this case, it doesn't. And the reason it doesn't is having 60,000 other measurements, at least 1 or 2 of those is going to be really highly associated with the blood pressure measurement that you don't have ahead of time. There were a number of other sensitivity analyses, but since I did not do my homework and I left in the slides that actually had the answers here, I'm certainly not going to poll the audience that there was differences in time and risk and dose as well. So let's talk a little bit about how impactful reliable evidence may or may not be, and kind of the journey ahead that we have all together to increase that reliability. So immediately after George published this paper, Paul Welton, who is one of the senior authors on the ACC guideline, wrote a commentary which basically completely dismissed this research. It says, yeah, George did a study and it was observational, but we know that prior observational studies and meta-analyses of very poorly done observational studies have said that chlorothalidone is superior. Well, that's not totally true. There actually was a prior study, observational study, that said it wasn't superior, but even I'll admit it was bad. It was a case-controlled study, and even I will make that plea to not use a case-controlled study design. They go on to say that, despite you doing a lot of work, this is still an observational study and it disagrees with what we believe, so it's not hard to imagine that your result is still just residual confounding. Even though there were 76 negative control experiments that showed that the residual systematic error was pretty damn small. So it's no wonder people really should be taking hydrochlorothiazide. Only randomized controlled trials will I ever believe. And I will only ever believe them because strong prior evidence, basically meaning the potency studies that were done in that laboratory, show that this is better. Why don't we all just wait for the VA trial to end? Okay. There are certainly some differences between the observational study that we did and the DCP study in the randomized controlled trial. First thing you keep in mind is DCP, this is an underestimate, was about $10 million. And we have an open community. It is really free. How much did it cost for us to execute Legend? Well, except for donations from places like IQVIA, thank you, Christian, it didn't cost me anything, not nearly $10 million. DCP's looked at the randomization of 13,000 people. We had close to 750,000. There's often this argument that the follow-up is a little spotty in observational data. Well, the follow-up in DCP was set to be 3 years, and the maximum follow-up that they could possibly afford was almost 5 years. And yes, average follow-up in the observational data was slightly less at 2 years, but there was a large fraction of individuals that we were actually able to follow 16 years out for long-term outcomes. 25% of them had over 4 years. And I think the most important difference is DCP wasn't even answering the guideline question. It wasn't answering the question, what drug should somebody start on? It was answering the question, if I'm already on hydrochlorothiazide, should I switch to chlorothalidone? And I understand the limitations on why it was set up that way. That's purely cost. It would be almost impossible or take a really long time to recruit treatment-naive individuals. Right? But in our target emulated trial, we were able to emulate the real question that the guidelines were trying to answer. I have just been diagnosed with hypertension. I haven't started a drug yet. Which one should I do? Well, as George mentioned, he stole my punchline. We were coordinating so much. DCP finally was reported out last December. That was super exciting, except not so exciting, because if we look at all of their primary outcomes, so MACE, heart attack, stroke, and heart failure, the DCP results matched almost number to number with the Legend observational study. And DCP actually only reported out one safety outcome. They reported out hyperkalemia, as would be expected, and they didn't even give a point estimate. They just said, oh, it's super significant. By the way, the size of the p-value is not related to how significant something is. It's either significant or it's not. But in this case, yeah, that's a pretty big ratio. Now, part of the problem we have is, great, DCP was published in the New England Journal of Medicine. There's no way the New England Journal of Medicine at this point is going to publish an observational study. So the problem that I'm purporting to this community and a bunch of people who are just getting engaged is we have reliable evidence. It, in certain cases, can emulate the trial that we want to do. Many of these trials can't be done. How can we improve the reliability? How can we improve? Totally spaced on that point. How can we increase our impact of the reliable evidence that we are generating? So I have 2 minutes for that discussion. I only have 1 minute for that discussion, but it was going to be a discussion time. How can we improve that impact, right? It's not just publications. What's going to help? Yeah.
Sorry, I just have a question. So if we have a lot of speaking work with observational data, and the previous lecture kind of talked about how we can overcome some of the limitations of RCT,
Yeah.
not having an RCT, but
Yeah.
using, so like with some of the tools that we were talking about before, with like propensity matching and the causal inference, are those good enough to match like a big VA RCT?
They did. What? Oh, how to summarize. Using best practices in terms of causal inference and observational data, right? Can we replicate randomized controlled trials? There certainly are examples, but there are also one of the problems we're facing is there are many counter examples because there is a vast literature of what I would call very poorly performed observational studies. So I don't think the hurdle that we're trying to overcome is, can we develop methods that adjust for residual systematic error so I can believe the estimate? I think the answer to that is, that's kind of a solved problem. The problem that we have is in the before times, right? Before one would try to do best practices with large-scale adjustment and actually estimating what the residual systematic error is, there's a huge body of really crappy literature out there. And that has kind of led to, at least as a clinician, sort of the knee-jerk dismissal of an observational study. And so how can we get over that knee-jerk submission? I mean, rejection of it. Is it working with clinical societies? Is anyone here engaged with a clinical society? Yeah. I mean, where might be the questions are the most receptive fields, clinical fields? Are there some places where what we've been doing has been more impactful? Yeah.
Is there a cost difference in the drugs?
Often there is.
I'm saying like the project which say,
Yeah.
if insurance is paying.
Yeah. No, it's yeah.
So
yeah.
That can be a selling point.
For those of you that didn't hear, this is a very good point. Showing comparative effectiveness is one thing, but saving someone money, if there is a cost or utility difference, is a direction that I think we as a community need to move. And it's important there to remember that cost differences are not just in relationship to effectiveness, but perhaps the largest cost differences come in terms of relative safety. Two drugs are going to be on the order of the same price and probably have the same effectiveness if they're in the same class, but can have very different long-term side effect profiles, which could have very different long-term costs, right? So bringing in the health economics is a great suggestion, especially when you start interacting with large groups like CMS. Yeah.
I think one area that is very important is the standardization, setting up standards for quality
Yeah.
and evaluation of observational data. All these societies have guidelines.
Yeah.
And there's guidelines, there's methodology teams that do this. But I think, having these standards about if we have an observational trial, how strong is the evidence? I think those are things that go in, because people look at the guidelines, right?
Yeah. Yeah. So this is a very good suggestion. Is providing guidelines on what represents quality and reliability in observational work. And that's kind of built on, the professional guidelines themselves. Almost all of the recommendations are graded in terms of how important they are. Like, a class 1 recommendation, thou shalt do the following or you will get sued, and it kind of goes down. But in terms of the importance, they're also in terms of the quality of evidence. This statement here was rated a class quality, I can't remember what was the term, but it was a grade A, which is the highest quality evidence based on a systematic review, right? But that systematic review was actually an indirect network analysis to compare two drugs that have never been done, hadn't previously been done head to head. We need to provide some sort of, kind of guidance on how you assess the reliability of an observational study. That's a good suggestion. That is. I've gone over time, and there's a slight wrap up. So thank you for your input, everybody.
Okay, we made it. I know it's really cold and you're probably getting hungry for lunch, so we'll keep this very short. We went over a lot today, and I don't expect anybody in this room to have absorbed all of it. It was kind of like a breeze through, and there are lots of opportunities to learn more. So if something you saw today was exciting, there's definitely tons of material where you can dig in, and I'm going to just highlight a couple things of where you can learn more. So first, we have something called the Book of OHDSI. This is freely available online. That you can go and peruse, and it talks through a lot of these concepts that we work through today. So if you're the type of person who really likes to sit down and read through it, you can do that with the Book of OHDSI. If you're like me and like a physical copy of the book, you can actually get it on Amazon. This is not a plug. It's at cost on Amazon. But if you like a physical copy, you can purchase it there as well. So this is a great resource. I have one on my desk, and I'll use it from time to time to really kind of state like things in a concrete way. Another tool that is freely available is something called the Eden Academy. So there was a project in Europe that was looking to convert lots of European data sources to the Common Data Model. And one of the outputs from that project was creating an academy. So you can go for free, sign up, and classes like we had today are available through that tool in a kind of online structured way that you've probably seen before if you've taken online classes. So there are great in-depth tutorials about the CDM, about Atlas, and various other things. So great, more structured learning there. And everything that OHDSI does, we try to record. So today we are recording this session, and then it will make its way onto YouTube. So OHDSI has a YouTube, and you can just kind of search around for things. One of the things I really like on our YouTube site is we've done like 10-minute tutorials. And so a lot of the key things will have these like kind of like 10-minute snippets. So if you're kind of just trying to get up to speed on like, how does Fee-Valuator work? Joel's done a 10-minute tutorial on it. So definitely check out the YouTube. We also try to make public what we're doing. And so if you're at the stage that you're thinking, hey, I'd like to get involved with a study. I'd like to see what other people are doing in terms of evidence generation. We do have a site that kind of combs through our GitHub repository. So if I was starting a new study and it's a public study that I want people to engage with me on, I would use our OHDSI GitHub repository to kind of say like, hey, here's my study. And what that allows is like there's this kind of crawler that will say like, oh, I see a new study here and Erica's running it, and this is like kind of all the attributes about it. So for example, here's a study that's ongoing right now by Chan Yanover, and what Chan was really interested in is understanding inflammatory bowel disease in patient cohorts. And so he's coming at it from like a clinical perspective. I'm at Johnson & Johnson. We're interested in the same things from our organization's perspective. And so we jumped on board and said like, hey, we'd like to do this along with you to see if we could learn anything that's of interest to our organization. And there's tons of other collaborators that are working with him. This is not the only ongoing study right now. There are lots of things that you could get involved with if you have your data converted into the common data model. And lastly, like at the beginning of the talk, I tried to stress that this is really to kind of give you an overview, and I hope today maybe something sparked your interest in terms of participating in the community. So maybe you want to focus on data standards. Maybe you're really interested in methodological research. Maybe you want to start and you're a developer and you'd like to get part of the open source development. You don't even have to have your own database. We have free databases like with like fake data that you can start developing on. You don't have to be part of a big institution with its own data. And maybe you just want to generate evidence, you have really good questions. These are all different places where you can participate. So yeah, if you would like to join, it's very, very simple. You just go to the OHDSI forums and introduce yourself. And that's the bar of joining this community. You don't have to do anything special. There's no money, there's nothing. You just kind of join by commenting on the forums. You can then start to get familiar with the community by coming to OHDSI meetings. Everything is posted online so you can see when we meet, but we do meet on Tuesdays on the East Coast time around the lunchtime. That kind of helps straddle both the Europe and the West Coast, and there are some Asia Pac centric meetings as well. I believe they're in the evening time for the East Coast people. You can join a work group. There's a form online that you can just fill out and then get connected with work group leads. You could start thinking about converting your common data model and just reach out on the forums if you need help to do so. Or you can just start by engaging in a network study. So I hope both yesterday and today we're engaging to you guys and you would consider still continuing to participate in the OHDSI community. So thank you so much. And I know we're gonna have lunch. If I can ask you for one more favor, there's this QR code. This will take you to a Google form. If you could provide us like a few minutes of feedback, I'd appreciate it. If you're so hungry and you can't do that, that's okay. We will send out an email so you could do this at a later time. But I figure it's top of mind if you want to complain about us, so you could do that now. So thank you guys so much. I know this is a really big room and it's cold in here, but you guys were great students today and we really appreciate your time. So thank you.